Oh, cool.
I wanted to get everyone to drink the fish right here. Because I have a lot more worthwhile out there. But nobody would. So I could be a witch for 3 minutes. I love that cartoon too much.
Yeah.
It really teaches you them how to regulate emotions.
We got plenty of music.
Not what I meant to do. All right. We're waiting on quite a few people to show up. I guess they say the traffic. I don't know. I didn't have a problem getting in here, so I don't know what's going on with other people, but I'm gonna go drop my food off in the fridge and then be right down. And literally in a couple minutes, and we're gonna get started as soon as I get back. Other people are not.
Just found out that they sent the emails I yesterday, I didn't see them.
Yeah.
You know, that's somebody sent an email saying you're approved to log in. So,
Actually make one changes on that. To make that one. When I was working at the exact same job already, being an LPN or NG.
you're not in your own now?
Huh?
Are you working on your
Yeah, I was working at their trauma center for a little over a year.
own?
And, but being an LPN on an RN treatment, she's. I had a patient. He was from me, but trying to do that stuff. And then.
I found out like
Last week.
about 11 years, not just involved in the whole Pennsylvania B12. All right, this is the meeting.
Yeah. Switch button.
Oh, recharge.
All right, make sure that we are signed in. We're gonna get going with our lecture today. It's a lot of information. And this is one of those chapters that me leaving it up to you guys to like peruse the slides or just have the lecture playing in the background. You are not gonna get the information that you need from it. So we're going to do it this way, so we're not missing out on any of the stuff. We're gonna do it this way, and if this works okay for you guys and you like it, then we will do chapter 17 and 18 the same way. However, unit 4, we won't be doing this way because unit 4, we need you guys to do your presentations during that time. So that won't be an option for unit 4. But hopefully this will be helpful for you guys for chapters 15 and 16. The immune system is a topic that is severely undertaught in the sciences because it usually requires background in chemistry. And usually it's taught initially introduced in microbiology, and then you have a separate class called immunology that you would take as a 3000 level class. So most of you guys, aren't going to be seeing that if you're going into nursing and getting just an associate's degree or something like that. So, so we don't see it a lot because it requires a lot of background understanding of biology, and we're only going to introduce it in two or three chapters. So it's a lot of information all at once, but we do have to have at least a general understanding of how this stuff works. It's going to feel like a lot. It's going to feel like, wow, you said general understanding. And I mean, quite literally, this is not even the tip of the iceberg, folks. So I'm giving you the most watered down version of the stuff that I can, but it's still going to feel like a lot. So go ahead and make sure that we're signed in. I'll give you another minute to get caught up. And we're going to jump into chapter 15. Chapter 15 is going to be discussing the innate immune response. Basically talking about how your body recognizes things as foreign. So there's 3 stages to how your body defends against microbes. The like primary immune response, secondary, and then like the third stage. The primary or first like immune defense level has to deal with like your physical and chemical defenses. That's going to be like your skin and stuff like that, having acid in your stomach. Those things might have other functions besides fighting off microbes and protect you from that, but they do help in keeping those guys out. This includes the entire 100% reason that you even have nose hairs, is to keep things out of your respiration. So stuff like that, we're going to be talking about that stuff in that first side of the response. The second part of the response is your innate immune system itself. And so that's going to be talking about your general immune cells and signaling molecules that are involved in basically responding to something foreign in the body. That'll be your neutrophils and your mast cells. That'll be your inflammation process and the fever and stuff like that. That's your body's ability to say that there is a person here that doesn't belong here, but I don't know anything more beyond that there's a person here that doesn't belong here. And then the third level is going to be chapter 16, which is our adaptive immune response. And that's going to be your B and your T lymphocytes. Your B and your T lymphocytes can tell which person is in here, and not just only which person, but they can say, you know, okay, this is not just Barry, but this is Barry's eyeball. We could say, okay, this is not, you know, just Megan, this is Megan's hair or something like that. So we can actually be very specific about which invader and which part of the invader. And that's going to be talking about our antigens. And so that'll be chapter 16 stuff. But first, chapter 16 stuff, the B and the T lymphocytes, they take a long time to get them up to numbers that can actually defend you adequately. Because like we've mentioned previously, you only have one cell that can see, you know, Barry's eyeball. You only have one cell. One cell. I don't mean one set of cells. I mean one single cell. So we have to find that one cell and then wait for that cell to multiply up and then do their jobs in your immune response. That takes like a week for it to be able to be activated, found and activated appropriately. So this innate response is really important in keeping things just under control until we can get the lymphocytes in place. I call our lymphocytes our B and our T cells. I call them our bombers and our tanks. I don't know how appropriate tanks really is, but B cell, calling them the bombers is really quite appropriate. When you're going to war and somebody's, you know, dropping bombs on another country, let's say, the bombs are doing the damage, not the actual bomber, right? And that's the same way with our B cells. The B cells are not doing damage. They're not really doing anything active in the fight except making antibodies, which are the bombs. And the antibodies have a ton of things that they can do in our body to help defend against things. And so we'll talk about that in chapter 16 as well. But again, we can't make the antibody until we find the B cell that recognizes that antigen, let it multiply out, and then it can start cranking out antibody. And that takes, like I said, a week. And then whatever time it takes for the antibodies and the T cells to clear the infection out to actually make you feel better. Most of the time, whenever you are sick with something, a lot of it is just waiting with this innate immune response going on until we get our B and T cells up. Because once we get the B and T cells up, it's a matter of like a day or two and you'll be completely 100% cured. But once you have the memory from that, the memory of your B and your T cells is so good and effective that it's the whole reason why if you have been immunized or exposed to something previously, you have proper memory, strong memory built up, that you could see that thing, it could get into your body, but you would never have any signs or symptoms of having been exposed to it. It's why me having had the chicken pox, I can walk up to a kid that has the chicken pox and not get any symptoms or anything like that. Because once you have the memory, you don't have to wait on that multiplying out thing to happen again. So that's the whole goal with that stuff. All right, we're getting signed in still, that's okay. And this number will always be at the bottom of the screen. So we're gonna go ahead and get started with our lecture today. These are our learning objectives. Just a reminder, they're here in the slides. So one thing I want to say about our learning objectives, this is all of them for chapter 15 on the one slide. When you just look at this list, the list itself doesn't look that big. And so one thing that I want to remind you guys is, you know, I'll say summarize the first line of defense against pathogen infections or whatever. And some of us are building our concept maps or making our outlines, and we're actually addressing what the learning objective is. But if we're just like putting a definition of something, that's not actually addressing the learning objective. But for now, already by now, if you've been turning in your concept map, so I'm not picky about the concept maps. If you're submitting a concept map and it has the general information on there, I'm only picky if you're just like listing some terms. I have a huge issue with just listing a couple terms, okay? But if you're actually putting in some effort to like have some explanations on there, most of the time, almost 100% of the time, really, I'm really close to 100% at this point. If you have info on there, whether you had ChatGPT make it or not, I don't get picky about it because I'm just saying, you're getting the work done and you're submitting it and you're probably getting full credit for it. However, when it comes to the test, what I expect you to be able to do is, for example, explain how white blood cells migrate from the bloodstream and into tissues. Not just say, oh, that's diapedesis. No, explain it to me. And like I said, when you guys are studying, I expect you to explain that to me without referring to your notes. By the time you come to take my test, you should be able to explain that from your memory without referring to your notes. That's what I expect when you get to my test. So if you can actually do these things, like recall the term for white blood cells or differentiate granulocytes and agranulocytes, explain the function of interferon in viral infections and cancer. If you can actually do that before the test, I promise you will be so much better prepared for the test. If you've already been doing that, I'm not talking to you. That's all I'm going to say. And if you're like, that's too much work, I don't want to do it, and you're fine getting the grades that you've been getting on your test, that's fine too. You do you. But if you want to do better, you have to do better. It's not just going to happen to you wishing. You can wish in one hand and do what in the other and see which one fills up faster. Yes, exactly. Right. I'm just saying, don't sit there and wish, do the work. All right, so moving on. Major components of host defenses. Again, first line of defense, second line, and third line. First line of defense is things like your eyelashes, your nostril hairs, your keratinized epidermis, the acid in your stomach, and stuff like that. The enzymes in your saliva, the tears, the moisture that causes that to run. Even your urine trickling out of your urethra helps flush out microbes. So all of that falls into that first line of defense. Second line of defense is going to be your actual activity of your innate immune system. So here we're talking about your inflammatory response. We're talking about the complement cascade. We're talking about neutrophils, basophils, eosinophils, and we're talking about mast cells. And then fever as well. And all of them have function and purpose, especially like phagocytosis is probably the most important thing as far as setting up for the third line of defense. You can't have the third line of defense without the second line of defense. So the whole point of how your B and your T cells even get activated is that your T helper cells, which are your captains, and they're the leaders of the other responses. You don't get antibody without the captains go ahead. You don't get your cytotoxic T cells that kill infected cells. You don't get those activated without the captains getting activated. And so we have to activate the captains, the T helper cells, but we only activate the T helper cells with the help of infected cells or antigen presenting cells. And so the antigen presenting cells are the ones that activate our T helper cells. They are phagocytes, or our P cells can activate as well. So we'll talk about those as well. So it's all these roles that all these cells can have, and some of them have more than one role. And yes, you need to know what all the roles are and how they incorporate within everything. But we can't activate our captains, our T helper cells, without the phagocytosis process of macrophages, pretty much macrophages. There's other cells that can be antigen presenting cells, but by and large, our most important ones will be our macrophages. We're going to learn about them in the innate response. All right, so we already have a question coming up. You're going to drop a little pin. Where on the chart would nostril hairs fall? Oh yeah, you have to kind of zoom in on it and move it. Then tap or click on the area. All righty. So yeah, we got first line of defense. That's absolutely correct. More specifically, the physical barriers. There is a physical barrier within that first line of defense. It's physically blocking things from getting into further parts of your respiratory. So nostril hairs will be in that first line of defense here. All right, other chemical defenses, we mentioned the acid in your stomach that prevents most microbes from being able to survive. If you're an endospore, you can survive the acid in the stomach quite easily. So that's often why a lot of the probiotics that you take to help with gut biome, probiotics are often endospore forming bacteria. And you've probably not even thought about it. If you look on the back of those bottles that are probiotic, it'll say bacillus something or lactobacillus something. And it's usually what those organisms are based off of, are endospore forming guys. Not all of them, but a good majority of them. In the intestines, we have digestive juices, including like bile. That bile itself is antimicrobial and it is alkaline. So we've got that shift from the acid in the stomach to the alkaline bile. And bile is released in a concentrated form, and that can be harmful to bacteria. So this is already a pretty hostile environment for bacteria to be able to survive in, but we know bacteria can survive in it. Other chemical defenses, semen has antimicrobial chemicals and vagina has an acidic pH. That acidic pH really in the vagina is maintained by lactobacillus bacteria in the vaginal canal. So bacteria are the ones that are fermenting in the vagina that is causing the vagina to have an acidic environment. Your body doesn't make the acidic environment, the microbes in your body do. They're so pretty important for maintaining vaginal health. Other things, lysozyme is an enzyme that basically breaks down peptidoglycans. This is a pretty key one to understand about and be aware of for the test, lysozyme. We find it in tears and saliva, and it, again, breaks down peptidoglycan, the bacterial cell wall. Resident microbiome can also have an effect, whether it's on the skin or in the digestive tract or in your sinuses, whatever. Number one, they take up stadium seats. Number two, they use nutrients in the area so the bad guys can't. Number three, they produce antimicrobial enzymes and things like that that basically can fight off any bad guys. They also help to create, like we said with the vagina, the proper pH in whatever environment that we're in. But they also can teach our immune system on when and where and how we're supposed to respond to things. Obviously, if you have a gram-negative bacteria in the gut, we're hoping that our body doesn't go haywire and have a freak out session from seeing bacteria in your gut because you're supposed to. So training your body when and where to see those pathogens largely depends pretty heavily on your resident microbiome. And we've talked already about this a little bit, but the flushing of the urine through the urethra, as well as vaginal secretions, the mucus and other things like that in the vagina help to flush out microbes in our body as well. The mucus, as well as other moisture in the respiratory tract, helps to catch the microbes. We have ciliated epithelium in the lungs that helps move like an elevator. We call it, I think it's called the ciliary tree or something like that. But it helps move constantly. Whatever's in your lungs out, and it's always doing that. And so the cilia in your lungs are helping with that. We'll learn about a disease in Unit 4 called pertussis. If you haven't heard of pertussis, the more common name for the disease is whooping cough. And so we know that the Tdap vaccine has a P in the Tdap. It's tetanus, diphtheria, and pertussis, right? So pertussis is protected from the Tdap vaccine, but we also know, like, you might have gotten your vaccines when you're younger, and then if you step on a nail or get a cut or something and go to the urgent care, they'll be like, have you had your tetanus shot in the last 10 years? And if you have, then you're fine. But if you haven't, then they'll want to ask to give you a booster because tetanus, right? But that's also when you can get your booster for pertussis and diphtheria. We don't have a lot of diphtheria in the United States, but we do have pertussis. And it's less severe in older people who have otherwise healthy immune systems. You still get cough and stuff like that, the whooping cough, but it's really, really severe in little babies. And so that'll be evident in Unit 4 when we get into respiratory diseases. There's a video of a little baby that has whooping cough. But if you've ever like breathed in your drink and had that cough, like you're just like gasping for air, that's the whoop of whooping cough. And I want to be very clear about this. Whooping cough was also known as the 100-day cough. They're not exaggerating with it. You have whooping cough anywhere from 2 to 3 months of coughing like that, nonstop. Little babies get this because babies can't be vaccinated. So we're talking about newborn all the way up to whenever you could get, a vaccine and have an immune system. So about 18 months. So then older folks that could be carrying the disease and having very minor symptoms that haven't been vaccinated come and meet their little newborn grandchild, and then the grandchild gets whooping cough and then dies. Right? So we're talking about a 100-day cough where you can't breathe. If you were coughing like that for 100 days, I don't mean you cough a little bit like that and then have a break. No, It's constant whooping cough. And so little babies can't survive that, number one. Number two, coughing like that damages the cilia in your lungs. And now you can't move the fluid out of your lungs. And so the cilia get damaged, and then what happens? You get secondary infections. So even though you might get over the whooping bacteria, now you're susceptible to pneumonia from other things as well. And that can continue on if it isn't stopped appropriately. So that's a huge issue. So that we actually rely pretty heavily on the ciliated epithelium in order to keep moving things out. Any type of coughing disease can cause damage to that ciliated epithelium if it goes on long enough. When we have pneumonia, we're talking about enough fluid gets built up in your lungs that now the ciliated epithelium can't manage it. It's just more fluid than it can manage. And then the bacteria basically form a broth out of the fluids and secretions in your lungs and grow in that. And so all of that might not be managed well. But if we can, it works pretty well, keeping things out from causing disease in there for the most part. Yes, innate immune system. All right, mucous membranes. Again, they're mucous membranes themselves. We don't have the dry, rough, constantly shedding skin cells like we do on the epidermis there. But in your mucous membranes, we're moist and we're permeable. So you're more susceptible to disease pretty much anywhere that you have a mucous membrane. But we have flushing of fluids there. We have special immunoglobulin that is associated with your mucous membranes. And we have the mucus itself to trap things. So there are ways that the mucous membranes can protect themselves as well. Hair follicles, we're shedding skin associated with them. We're shedding the hairs themselves. And then skin glands, of course, making sweat and other things to bite off. The keratinized skin layer is probably one of the more important aspects of the stratus corneum in your epithelium, in your epidermis. And it's a waterproofing protein, keratin is. And so it creates like a barrier that microbes just literally it makes it hard for microbes to get into. And we know this is true because if you were like out and about in the normal bacterial environments with normal skin on your body, nobody's really worried about you, are they? But you get a burn, you get a 3rd degree burn, your whole arm, let's say. Oh, the doctor's worried about you getting infections? Yeah, because you don't have that keratinized layer on you anymore to protect you. And you've got this moist skin underneath. That keratinized barrier is gone. Then we're worried about infection now. So this actually creates a very helpful and significant barrier against microbes there. So keratin is what is doing that for us. So that's pretty much it for our primary guys. We're going to move right into the immunology of things. Okay, so immunology is your body's ability to recognize self versus non-self and react to self versus non-self appropriately. That's the idea there. And the main goal of it is to destroy foreign molecules. White blood cells are known as leukocytes. That is a term that you have to be aware of. Leukocytes. This is important that we know the difference between a leukocyte and a lymphocyte, right? So leukocytes are all of your white blood cells. White blood cells, leukocytes, interchangeable term. Lymphocytes will be your B and your T cells, okay? And they're part of your adaptive immune response. All right, so our leukocytes, our white blood cells, they are trained to recognize self and non-self. And they have markers on their surface that helps them identify that. The things that they can see as non-self, we call them antigens. It's a very broad term, but there's like specific antigens and then general antigens, okay? So we know antigens, also known as immunogens, are any molecule that the body could see as foreign and react to as foreign. We also know that if we can recognize self, we should be able to do that appropriately. We know that there's disorders that, sometimes we recognize self as foreign, unfortunately. And that's what's going to be leading to our immune disorders like rheumatoid arthritis. In rheumatoid arthritis, we have antibodies attacking our own joints, then inflammation in the joints leading to damage of the joints, and then the body trying to repair that damage, leading to basically scarring in the joints, causing the swelling in the joints and the dysfunction of the joint themselves as a result of that. And that's all because we saw self as foreign. And so that training mechanism is not perfect by any stretch of the imagination. Some of that is going to depend on molecular mimicry, so that is microbes that look like our molecules. But sometimes it'll be other things. And we'll talk about that more in chapter 17. Okay. All right, so antigens can basically be seen as general antigens, antigens that let me see that there is a non-self person in the room. You don't belong here. You're a person. That's all I know about you. Versus Again, saying that is berry, or more specifically berry's eyeball, that sort of stuff. So general markers, PAMPs, pathogen-associated molecular patterns, they're patterns. If you have this general pattern, we can say you're non-self. There are obvious markers that we can call PAMPs, okay? These will be things like lipopolysaccharide. Do you have lipopolysaccharide on your own cells? No. Do you have peptidoglycan as a cell wall in your own cells? No. Do you have in your cells double-stranded RNA? No. Only viruses can have double-stranded RNA. Only bacteria can have peptidoglycan, and only gram-negatives can have lipopolysaccharide. Now, if I see lipopolysaccharide, I know immediately you're not self. But do I know anything else about you? Do I know what kind of gram-negative you are? No, I just know you don't belong here. So PAMPs are very general markers that indicate to our body, we know immediately that you're not self. But that's all I know. So I know it's safe to eat you up and kill you. But I can't create memory or antibody or anything like that based on a PAMP, okay? Because it's super general. So these are very, very general markers on cells. Again, large groups. They identify large groups of pathogens. Pattern recognition receptors are the receptors on our cells that recognize PAMPs, okay? So usually our phagocytes will have these pattern recognition receptors, and those PRRs, those receptors will recognize LPS and peptidoglycan and double-stranded RNA. And there's other ones too, but those are definitely the big three that you should know about, okay? All right, so that's the PAMPs. Here's our next question, true or false? A PAMP is a molecular marker that helps the immune cells recognize general groups of microbes. So true or false? I forget, I have to click. All right, so this one is definitely true, good job. All right, major functions of the lymphatic system. The lymphatic system is there to filter things out. So this is where I get into the explanations of what is going on. So these explanations, as I go through these explanations in this class, you won't find this on any slides. So if you haven't been watching the lectures, this is the stuff you've been missing, okay? Not on my slides. I don't read from my slides in my lectures. So imagine that you are walking around and you stub your toe on the side of your bed or what. I do it all the time. And I have dog beds that are on these little stands and I stub my toe on it all the time. So let's say you stub your toe and you break the skin when you do it. A little bit of a cut there. Bacteria can get in that way, right? Okay. Let's say that happens. It happens all the time. Now we have created a break in capillaries in the tissue as well. Microbes can get into your bloodstream that way. So your body, those cells in the area have to send out signals to each other, and they are molecules that they use as signals called cytokines, which we'll talk about in a moment. So they basically are a call for help. They release these chemicals saying, I'm damaged, my neighbors are damaged, damage is happening here. Those signals basically build up in that area. Cells nearby get the signals. They send more signals asking for help. Cells in your bloodstream, like neutrophils, get the signals and they follow the trail of help, 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 and listen for the call and migrate to the area where you stubbed your toe. That's how neutrophils get called to the area. At the same time, because there's damage going on, your body's going to increase blood flow to the area so we can get neutrophils and clotting factors and things like that to the area, but also decrease blood flow out of the area because we don't want microbes to be traveling out of the area through your bloodstream. So vasodilators and vasoconstrictors will also be recruited in response to the damage that's being done. So we'll have that going on. And then our white blood cells get in there and our phagocytes start eating things up. We know about phagocytosis already. This is an idea that we have. Recognizing PAMPs, right? So bacteria that was on the bottom of your bed, because I'm sure that we have our rooms completely sterile all the time. But the bacteria that are in our environment got into our little toe, and now they're in there. We're recognizing the peptidoglycan and eating up the bacteria in the area in response to the PAMPs. So that's the idea of what's going to happen. Now, it's a problem if we just let all of those cells and the fluids that come with the cell, including the nutrients that help power our neutrophils, all of the fluids that come in from edema, from the swelling, from the inflammation, we can't just let it re-enter the vascular system, right? Because bacteria could get back in through the vascular system. Your vascular system, your blood, is sterile. It's an open stadium. There's no microbes sitting in the seats. There's nothing there. Are your immune cells there? I mean, yeah, so you've got some protection there, but we can't let microbes take up house in the circulatory system. So what happens is all the fluid and the white blood cells and all the junk that's in the area, as it's getting reabsorbed back in the body, we don't let it go into the bloodstream. It rejoins through the lymphatic system. Lymphatic vessels are all throughout your body, just like your blood vessels are. And they kind of mimic where they are in the body in relation to the blood vessels. So any fluid in your extremities, anywhere in your body that needs to rejoin the body after infection or inflammation or damage has to go through the lymphatic system. So that's the whole point of this. So it's a set of vessels and a mechanism of moving all that fluid and reincorporating it back into the circulatory system. But after we filter it through our lymph nodes. And what do we have in our lymph nodes? White blood cells. And they are scanning every single thing that comes through. So they're literally sitting there, your B cells and your T helper cells and other macrophages and everything like that is constantly surveilling anything coming in from an injured area before it comes and rejoins in the circulatory system. That's your body's mechanism of protecting from invaders that could come in through any sort of break in our security. Make sense? So it's the idea of the lymphatic system. All right, so we've got fluid. This is the fluid of your blood. If it's not a red blood cell, that could be part of plasma. It's part of the plasma, basically. So it includes your white blood cells and all the nutrients, the clotting factors, fats, all of these other components that could be in there inside of your lymphatic fluid there. And it's all the fluid that moves into the extracellular spaces in response to damage. The vessels move in only one direction, right? They move from the extremities to the heart. And the fluid pumps in that direction as well, from the extremities to the heart. So like veins. So we know arteries away from the heart, veins come back to the heart. Your lymph system only moves in that direction, not through veins, through their own vessels. So that we're not rejoining the heart without being filtered, okay? Lymph also is not part of your circulatory system. Again, they're separate vessels that basically move only the fluid portion that moved out into tissues in response to damage. If you were picking up, like I'm coming over here to get my drink and I accidentally hit my elbow on this thing here, already damaged, fluid's going to move into the tissue and whatever. Anytime you have any even remotely, even small little bit of damage, fluid moving out of your blood vessels, cells coming in to repair any damage that could be done. We cannot rejoin the circulatory system. We come back through the lymph. So it's happening all the time. You don't have to have serious damage, even minor damage. This is happening all the time with your lymph, okay? So again, lymph is not moved by your circulatory system. It's not joined that way in your circulatory system. It has to be fed back in to your circulatory system by the movement of your skeletal muscles. So your heart is not a skeletal muscle, it's a smooth muscle. Your heart does not pump your lymph. You have to move your body to pump your lymph. Now, can you manually massage your muscles or your skin or your tissues to help with movement of lymph? Absolutely. Is like gua sha and lymphatic drainage stuff that you see on Instagram actually effective? It is. It's not a gimmick. That's not a gimmick. It does help with lymphatic drainage. Now, how much that's going to help slim your face? That's unrealistic. But does it help with lymphatic drainage? Yes. Okay. It does. So lymph only gets cycled back in by movement of skeletal muscles. So whenever we're talking to our patients who have had surgery, we have opened up a hole and now they need to repair damage. All damage repair is guided by our inflammatory response in your innate system. All of it is. Any type of damage. I don't care if it was sterile damage that was controlled or you stubbed your toe. All of that inflammatory healing process is guided by your immune system, which is basically going to be moved and cycled through your body and replaced with new cells based on skeletal muscle movement. Otherwise, you're just going to build up fluid in the area and it's just going to sit there. And so are the bacteria, and so are all of the cells and all the waste and everything like that. So the only way you're going to get that cleared through your lymphatic system is by moving skeletal muscles, which is why the number one thing after somebody comes out of surgery or has been ill or sick and been not moving for several hours, what's the first thing you tell them to do? Walk. The number one most important predictor of somebody healing after a surgery is movement. And if they can't move, then we will put those things that squeeze their legs. You guys know what I'm talking about. They like use them now. If I had my tonsils out a few years ago, yes. And they put those things on your leg that like pulse, they squeeze your leg muscles. And that's to try to get extremity lymph back moving through so that you don't have to get up and walk right away. You can sit in the bed and recover a little bit, but still get that lymph moving. Well, what's the first thing they want you to do as soon as you can? Get out of the bed and move yourself, because that's even better. Get that lymph moving, get those innate cells and all those, all the waste from responding to the inflammation, all of that, get it filtered back in through the lymphatic system. Here's our lymphatic system in the green in this middle image here. We can see that it does overlay pretty well with our circulatory system, but again, it is not linked in. It goes from the extremities back in. It's just a system to help filter things back in before it rejoins your circulatory system. Okay. So here's a little quiz about it. Quiz question, multi-select, select all that apply. How is lymph transported through the body? I believe 2 of these is correct. Yes. No, not technically. So we'll talk about what an epitope is in chapter 16. Yes. All right. So skeletal move. So we're moving from the extremities to the heart because of skeletal muscle movement. Beautiful. Good job. All right. So our lymphoid organs, our primary lymphoid organs are where our cells come from. And then the secondary ones are where the functions are kind of performed. Okay. So all of our innate cells come from your bone marrow originally, and your T cells go through training in the thymus. That's the only thing that happens in the thymus is we're training our T cells. So once we make the T cells in the red bone marrow, they'll move into the thymus for their training. Okay, thymus is here. That's why I'm doing this. It's right here. And your thymus and your immune response is going to be strongest in your childhood years. Once you hit puberty, it's a decline from there. Okay. You think your immune system is the strongest as an adult? No. Your immune system is the strongest as a child. And so we often think, well, children get sick a lot. Yeah, because that's their first exposure to the things. Once you're an adult, you have the memory there, and it feels like you have a stronger immune system. You don't. You've been exposed to more things during childhood, and now you have the memory as an adult. That's what's really happening. But all of your newest, most of your cells all exist. It's the strongest. It's the most ready to respond to foreign things in childhood. So you don't get an immune system at all until you're about anywhere from 6 months to 18 months old. So that's why we wait for our vaccinations for 18 months. Most people will have their immune system fully formed and ready to go at 1 year of age. And so we push it a little bit just to make sure that the babies have an immune system. They literally don't have an immune system before that. We've talked about that too, right? They get their protection from the mother's breast milk. So that's a type of passive immunity that we'll talk about too. All right. Anyways, we get our white blood cells. We get our neutrophils, we get our T and our B cells and all of that, and they're basically going to be in your tissues or in your bloodstream, depending on what type of cell and how we're responding to things, all right? All of our lymphatic screening, as we're moving our lymph through our lymph vessels, all of our cells will be basically concentrated in our lymph nodes, and it's basically like a way for our body to have exchange of that interaction, communication. So as we're filtering all that lymph, and we have checkpoints at every lymph node, we have concentrated numbers of B and T cells, so we can have interaction between our T helper captains, between our T helpers and like the other cells of our immune system. That can happen in the lymph nodes. The B cells will often take up home there and like start making antibody from there. And so they'll multiply and divide and stuff in the lymph nodes once they're being activated. And then that's why your lymph nodes swell, because once a B cell gets there and it's been activated, it's multiplying like by the millions in that lymph node, and then your lymph node swells. Anybody know about how big a lymph node is and if you're healthy? How big is a lymph node if you're healthy? Yeah, it's about the size of this eraser, about the size of half of a pea. They're little. And some people argue with me. I'm like, no, mine are big. Yeah, you got allergies or you got an infection, you got immune response happening in them. That's why they're big, because you got B cells dividing in them. And I'm telling you, as a person that worked in the HLA lab, which we'll learn what HLA is too. It's MHC to be clear. But I worked in the transplant lab at Baptist, doing all the testing for transplant patients, and we had to take lymph nodes from the donor, the person that was donating the kidney, usually a brain dead person, right? The kidney or the heart or the lungs, and they would send us lymph nodes, and I'm telling you, they're so tiny, and we had to like get the B cells out of them. It was a chore. Getting something out of something this small. If they had inflamed lymph nodes, they had an infection, we couldn't use their organs, period, right? So I didn't see big ones. Normal lymph nodes, very, very tiny. Even the ones in your neck, They're normally tiny, okay? So lymph nodes. Lymph nodes are the ones, basically, they aren't just in your groin, in your armpit, or in your neck. We have lymph nodes everywhere, all throughout our body. It should be a concentrated lymph node numbers in our groin and in our neck and in our armpits, because these are areas that are basically major checkpoints for interaction with pathogens. I don't know what's going on. Anyways, salt, malt, and GALT. The ALT of these guys is associated lymphoid tissue. It's like tissue. I'll check this out. Oh, is it the wind? There's nobody out there. I think it's just wind. It's because it's so cold in the hallway versus in here, and then people going in and out the door. I think that's what it is. Anyways, ALT, associated lymphoid tissue. It's like lymph nodes, but not in node form. It's like a bunch of tissue that has a similar function to that. So this is skin-associated lymphoid tissue, mucosa-associated lymphoid tissue, and gut-associated lymphoid tissue. Okay, that's what those are. But now we have the spleen. Okay, the spleen is also part of our immune system. Now that's its secondary function. We know the spleen normally filters out old red blood cells and stuff like that. But while it's filtering the blood for old red blood cells or damaged red blood cells, it also has macrophages there that eat up those red blood cells, but that can also eat up any bad guys that it filters through the blood. It does not filter the lymph, it filters the blood. So if for some reason a pathogen did get into the bloodstream and didn't get filtered from the lymph, then the spleen is there to filter out guys that made it into the blood. So if you lose your spleen, are you more susceptible to infections? Yeah, you are. Can you live, if you have an otherwise healthy immune system, can you live a perfectly normal life without a spleen? Yeah, but you're still going to be at risk for more infection if they got into the bloodstream. Yes.
So we're talking about lymph nodes and stuff. So how does like cancer work?
Depends on the cancer. So if we're talking about leukemia and lymphoma, we're talking about the overgrowth of white blood cells there. Depends on the white blood cell. So if it's a B or a T cell that is overgrowing and that it's being unchecked and multiplying too much, and usually we're talking about way too much if we're talking about cancer, and that's lymphoma. It's a B or T cell doing that. And a lot of people think like lymphoma is in the lymph nodes. No, we're talking about B or T cells. B or T cells are multiplying out that much, and that's lymphoma because those are lymphocytes. Okay. Leukocytes, the other white blood cells. So we're talking any of the other white blood cells, neutrophils and basophils and eosinophils. If those are multiplying out of control, that's leukemia. Okay, and lymphoma is a type of leukemia technically. All right, so it's a subset of it. It's just a more specific subset. So that's a type of cancer that's directly associated with the overgrowth of those types of cells. Now, if you have breast cancer, right? So we might have heard about if you have breast cancer and we had to remove a lot of your breast tissue and stuff like that. If let's say you have breast cancer, we have an immune response to cancer cells. Your cytotoxic T cells, which we'll learn about in chapter 16, okay? They kill infected self cells, basically your own cell, your lung cell, your skin cell, even your breast tissue cell. Any cell in the entire body that has a nucleus can call out for help saying there's something wrong inside of me. And they reach out and call for help with MHC1. Okay? Something's wrong inside of me. And I found this. Something's inside of me that doesn't belong. Okay. I wish I had this facing more this way. I can turn the screen at least. So if I'm going to stand over here, at least the recording will get it. So if I have something wrong with me, I found this inside of me. I'm going to hold it out and say, help, I found this inside of me. Somewhere there's a T cell that recognizes this, lip gloss. Somewhere there's a T cell that sees this lip gloss. And it will be able to find it and say, I've been looking my whole life for that lip gloss. And then what does it do that we talked about in class? It's going to say, oh, you found the lip gloss inside of you? Kills the cell. You are not supposed to have something wrong inside of you. That's what the T cell says. And so you're gone now. Your body can sacrifice a lot of cells, and you have billions of cells in your body. We can regrow that cell and replace it. All right. So that's the idea. If you have cancer, you have something wrong inside of your cell. Now your body has to be able to tell the difference between that something wrong being self and non-self. cancer can lead to self-changing enough that it looks like non-self, and we can react to molecules inside and say, you are now non-self because something's going wrong enough in there. And we can kill the cancer cells that way. Many types of chemotherapy often target that. They target your immune system, trying to help your immune system out and say, help you find that better. There's a study that was recently published. It's so good. I'm so happy I get to say something good about this. There's a study that shows that if you are receiving that type of chemotherapy that targets your immune system and helps your immune system fight off cancer and see it as non-self. There's a study that was just published very recently that shows that if you're having that type of chemotherapy and you get the COVID mRNA vaccine at the same time, you are significantly more likely to fight off your cancer because it's basically helping your immune system react. That is gold to someone like me. We get all these poo-pooers saying about how bad a vaccine is for you, and it's been clinically shown that it is significant improvement. Now, are there side effects associated with vaccines that could put somebody who's already having issues with cancer and chemotherapy and stuff like that at risk? Yes. So we don't necessarily just want to reach for COVID vaccines when somebody is getting chemotherapy, but it might turn into a possible route of helping amplify the response of the immune system against cancer. Really cool stuff though, right? All of this is your immune response. And your body has a mechanism to fight off cancer built in with it. And it's the same mechanism that fights off viruses. It's non-self inside stuff. And so there's that. The problem is then when you have cancer, like we're saying here, right? Breast cancer, for example, your body is filtering that stuff out from the lymph and saying like, hey, this is, I found this hanging out outside of the cells, or I found this where it didn't belong, or this cell has something wrong with it. It's a non-self thing. And you have that being filtered out in your lymph if those cells no longer stick to where they're supposed to. So we know breast tissue is supposed to stay in the breast tissue there. When you have cancer, we start losing our ability to adhere to our neighbors because there's a lot wrong with a cancer cell. So when they stop adhering, where's it going to get filtered out first? Lymph. And so then the lymph node, they get stuck in the lymph node where it's interacting with the B cells and the T cells. That's the first place where you're going to see cancer metastasize for any type of cancer. Does that make sense now? When we put it that way, because your body's still filtering out as non-self and it's still getting interacted with as non-self. So the first place that you're going to see a non-self thing get stuck and migrate to is through the lymph. We can't go through the bloodstream typically. I mean, can you technically? Yes. So that's how we get metastasis to other tissues is through the bloodstream. We're tracking? That's a good question. We're thinking beyond a little bit, but it does relate directly to all of this. Yes.
I read a deal not too long ago that they said that Manjaro is doing the same thing.
For what?
Cancer and some multiple other things.
Trying to help with the immune system and
Yeah, like getting rid of certain cancers.
yes. So this
Like
is
shrinking those particular cancers.
so this is a field. This is a whole field of medicine that I feel like is not talked about a lot because it requires understanding of the immune system. You can't understand what chemotherapy does unless you understand the immune system. So whenever you've ever heard of somebody getting chemotherapy, it's all immune system. All of it is. It's all targeting the immune system and trying to get it to work. Anybody ever. Your mom had chemotherapy, it's this. It's getting the immune system to react. That's what it is. And so there are other chemicals that might be there that help stop the cell cycle growing as well. But they're always working together. They're never working alone. So drugs that might stop cells from growing, we don't just want to stop your cells from growing, right? Because that stops your own cells from growing too. And that's why people lose their hair. Because whenever you stop cells from growing, you can't replace hair follicles. They just fall out and die because the cells die. So that happens all over your body when we stop cancer from growing too, if we use it all throughout your body. And if we're using it throughout your body, that's because you're in stage 2 or higher typically. Do we use it in stage 1? Yes, but that's also because we don't think you're just in stage 1. We think there's a possibility you could be higher than stage 1. We just haven't caught it yet. So the likelihood of there being one cell that escaped, it's too high. We usually don't want to take the risk, right?
What alopecia is that? Like
Alopecia, it can be related to this, but it's a little bit different. So then we're getting into autoimmunity there. And so when we talk about that in chapter 17, we're going to talk about how the immune system, again, something's wrong on the inside. I found something wrong on my inside. Help, right? So if we see that going on and our cytotoxic T cells come to target and kill you, now imagine that it's a healthy cell, but we targeted it wrong because we saw anti-self reaction with autoimmunity. That is what type 1 diabetes is. So in type 1 diabetes, the T cell sees the beta insulin producing cell of the pancreas. It sees those cells as foreign and kills them all. And now that person can't make insulin anymore. The more you know. Okay, so we're here now. We're there now. It's meta. It's a lot more meta than you probably ever realized it is. That's why we're taking time on these chapters. Okay, we getting there? We're thinking. We got the juices flowing. Good. All right, so we'll click through this stuff. We did this. We talked about the thymus. Again, it's just an organ where the T cells mature. There's not much else to say about it. It shrinks pretty much once you hit puberty. Lymph nodes, we talked about them. And we talked about the enlargement. We talked about the spleen. We talked about the associated lymphoid tissue. Okay, so whole blood. If I take the blood directly out of your body into like a red top tube or something as a phlebotomist, then we have your whole blood there. That whole blood, and I let it clot. The fluid that's left after the clotting is called serum. If I added something to the tube to keep it from clotting, so this is your green top tubes and certain other kinds of tubes too. If there's anything added to it to keep it from clotting, then that liquid that we get when we centrifuge it and separate the cell, the liquid from that is called plasma. So if there's anything added to prevent clotting, the liquid is called plasma. If there was nothing added to prevent the clotting and the clot happens, and then we separate it, the liquid is called serum. There is a difference. And it absolutely does matter whenever you guys get into medicine, because the lab will call you repeatedly and complain about it if you don't get it right. It's okay. We needed serum. So ever ordered a serum pregnancy test or something like that, we get a red top tube. It comes to the lab lets it sit there for 30 minutes because it has to clot. It has to. Once it clots, they spin it in the centrifuge and they take off the clear fluid. And what do they do in the lab when they do a serum pregnancy test? Do you guys know? We take a pregnancy test card like you would pee on, but we put the serum on it. It's the exact same test. It's just, if we measure the HCG level, that's a different test. But if we're just looking for positive or negative, it's like the P test, but we're using serum instead. Kind of cool stuff. Yes, question.
You're talking about that serum.
Yes.
The other day I had to take my dog to the vet because he had a massive knot right here. He did get shots about a month ago, so I didn't think it was that. But anyways, the vet sat there and she's like, it's just serum. So I mean,
yes.
I mean, she took like 40 cc's out of it.
So we could call it serum, we could call it plasma, but this is where we're going to talk about that inflammatory response and the fluid that comes from the inflammatory response, which we're getting to next. Okay. So our leukocytes, right? Our leukocytes, again, white blood cells. Yes.
Plasma, since it doesn't clot, do you have to spin it? Is that how you separate it?
Yes, we put it in a centrifuge and it spins it fast so that we can get the sedimentation of the red blood cells since it doesn't clot on its own. Okay, so white blood cells, also known as leukocytes. There are two types of white blood cells, the granulocytes and the agranulocytes. So I'm gonna come back real quick. Those are our white blood cells. We also have other blood cells that are not white blood cells. Red blood cells, for example, obviously not white, right? Red blood cells and platelets. Those are not white blood cells, okay? I think there's a slide. There is, okay. Yes, so those are non-leukocytes, red blood cells and platelets. Pretty much all the other ones are white blood cells, okay? So the granulocytes and the agranulocytes. Remember, agranulo means without granules. Granulocytes with granules. Okay, what are granules? Quite literally, if you stain the cells, it looks like there's little grains inside of the cell, little dots, little dark dots. This is often because of whatever the cells has. We look at this image here. You see the little dots inside of these cells? Those are the granules. You see how these other cells don't have those little dots? That's on purpose, okay? And it's literally based on what you see in a microscope, based on what your hematology lab person sees in the microscope, okay? And so basically, like when you draw a CBC and you send it to the lab to have them, they do the differential on it. And they tell you how many, neutrophils and how many lymphocytes and blah, blah, blah. You get that readout back, as well as your hemoglobin and all that. The person that does that test is the hematologist. Okay? And it's in the hematology part of the lab. There's a machine that does that, as well as if they do the differential, they look in a microscope at the cells. And they use a stain. When they stain it, if they see granules, it's a granulocyte. If they don't see granules, it's an agranulocyte. And there's other defining features. That's literally how this is defined. What are the granules? There's special little vesicles inside the cell that have like digestive killing ingredients. ingredients inside the cell to kill foreign things. That's what's in the granulocytes, okay? Now, do other cells have things inside of them to kill things? Yes, they do. It's just you don't see them in the stain. That is literally the only difference, okay? It's whether you see them in the stain or not, okay? So granulocytes, these are the fills, all of the fills. Neutrophils, basophils, eosinophils, as well as a non-fill, the mast cells. Those are the granulocytes. There's 4. Neutrophils, basophils, eosinophils, and mast cells. All right. The agranulocytes. There's 2 major groups of agranulocytes. There really is. I know it looks like a lot's going on over there, but don't worry about it, okay? The 2 major groups of agranulocytes are the lymphocytes and the monocytes and their derivatives. Monocytes, they are a type of phagocyte that is in your bloodstream. When it moves into the tissues, it differentiates in either to macrophages or dendritic cells. So again, monocytes are in the bloodstream. If it's in the tissue, it's a macrophage or a dendritic cell. Most of the work against infection is going to be happening in the tissue. Whenever you stubbed your toe and you opened up the wound on your toe and the bacteria got in, that's damage that's happening in your tissue, not in your bloodstream. And so cells come out of the bloodstream and move into the tissue. They become macrophages or dendritic cells. Now, how is it determined whether they become a macrophage or a dendritic cell? And what does that mean? You don't need to worry about that, I promise, okay? Not in my class, all right? If you want to take immunology and you want to get into more detail with it, we can. But it's signals in the area. Are we having damage in the skin or in the lung or in a liver or whatever? And then we will differentiate into a macrophage or a dendritic cell. And it's not just macrophage. We have lung-based macrophages called alveolar macrophages. We have liver-based macrophages, I think called Kupffer cells or something like that. They're very specialized to the tissue, and we have names for them for where they are. Focus on the general here, okay? So once we get into the tissue, we basically are honed to that tissue from then on. We can move out of it, but we'll usually come back to that tissue, okay? So that's our monocytes in the blood, and then they move out and become macrophages or dendritic cells. All right, the lymphocytes, the only ones you need to know, I know there's more on here, there's the natural killer cells and the gamma and delta T cells and all this. The ones I really need you to know about are the T cells and the B cells. They're the only ones I'm going to ask you about on the test, okay? T cells and B cells. We're not going to talk about their function now. We're going to talk about them in 16 here in a little bit. Are we tracking? Okay. All right, here's a question about them. Let's see if you really are tracking. Multi-select, select all that apply. Which of the following cells are granulocytes? It's a common type of question on my exam. All right, so the fills and mast cells, those are the granulocytes. So lymphocytes, macrophages, and monocytes are all agranulocytes. All right, moving on, the mononuclear phagocyte system. This is basically like the extracellular highway. So the way that cells get around in your tissues, they don't move through other cells. That's ridiculous. They move between the other cells. And that stuff that's in between, we would call it the extracellular material, right? The extracellular matrix basically forms the mononuclear phagocyte system. It's how our phagocytes move around in your tissues. Monocytes, again, gave rise to the macrophages and the dendritic cells. Monocytes are found in the bloodstream. Macrophages and dendritic cells are found in the tissues. They are phagocytes, okay? Cytokines, we mentioned this as well. These are signals that are sent out from your damaged tissue, as well as any responding immune cell at all. So basically, an immune cell gets there. They can send out signals when they respond to things. They can also receive signals whenever they're interacting with things. So those signals are called cytokines. Cyto is cell, kine means the signal, okay? There are 4 categories of cytokines. Pro-inflammatory. These are the signals that are basically favoring inflammation and healing and things like that. Anti-inflammatory. These are the signals that are to say we are done with the inflammation response. We don't need to keep up with it. Vasodilators. So you can imagine too, if we have anti-inflammatory cytokines that our cells can send to say, hey, stop with the inflammatory response, you could use those as drugs to modulate somebody's excessive inflammation. And we do. Okay. Pro-inflammatory, if we're trying to encourage immune response, can we send those signals in our body to help fight against cancer? We do. Okay. Vasodilators and vasoconstrictors to basically control blood flow to and from an injured area. And then growth factor cytokines. These are cytokines that are used to help initiate the growth and replication of our B and T cells. So again, if I only have one cell that can see Barry's eyeball, then we need to multiply that cell out once we find Barry's eyeball. And we want that cell to multiply out. And the growth factor cytokines are the ones that signal for that growth. We've talked about phagocytosis. Phagocytes are the cells that perform phagocytosis. We're finally here, you guys. We're finally here. The three main types of phagocytes. This is not an exhaustive list, but these are definitely the most important cells that are phagocytes. Neutrophils, monocytes, and macrophages. By far the most important phagocytes. Okay? There are other cells that can perform phagocytosis. Now, neutrophils. Once you stubbed your toe and the bacteria got into that wound, now we have cells in the area saying, help, there's damage. We have things like mast cells that are found in the tissue area that can immediately start releasing cytokines to call for inflammation and to call for other cells to move into the area. Neutrophils don't typically exist in the tissue. They exist in your bloodstream. In fact, they're the most numerous of all of your white blood cells in your bloodstream. So neutrophils aren't at the site whenever it first happened. But the damaged tissues and mast cells that are at the site they're calling the neutrophils there. So they sent out cytokines, pro-inflammatory cytokines. The neutrophils move into the area from the bloodstream. Now they're there, they can start eating up the bacteria. We see peptidoglycan, we see PAMPs, and we start eating things up through phagocytosis. They'll just eat things up and break it down. And that's all they do. So they get there, they're like our first line soldiers. They're just there to kill bad guys. Looks like a bad guy, kill it. Looks like a bad guy, kill it. Looks like a bad guy, kill it. We don't care about the information about the bad guy. We just know it's a bad guy, kill it. Okay? The monocytes and the macrophages. Once the neutrophils start killing stuff, the monocytes and macrophages get called in. So they come into the area, monocytes come out of the bloodstream, become macrophages, and now they're eating up all of the waste that the neutrophils left behind, as well as the dead neutrophils. So they're basically coming in and assessing the damage of the battle and saying, do we need to call in our captains? And they're getting all of the information, all of the evidence of the battle and collecting it. That's what our macrophages do. And they're eating it up, right? So also phagocytes, but the neutrophils are just phagocytes that ate and killed very non-discriminately. You're not self, eat and kill it. The monocytes and macrophages come in, they eat that waste, and they're collecting it so they can go call for help. Okay, so we're setting that up. We're going to get to chapter 16 and figure out what they do with that in a little bit. Okay, that's the idea of those phagocytes and their function in this. Neutrophils react pretty much to any infection. Eosinophils react to parasitic infections, but if your body isn't trained properly with parasites early on in life, which most of us living in first world countries, our bodies have not seen parasites early in life. All right, then you're more likely to have allergies. Eosinophils, basophils, and mast cells react inappropriately if they aren't trained properly with parasites. So allergies are, generally speaking, almost exclusively a first world problem. Third world countries that have parasitic diseases typically don't have allergies. We'll talk about that in chapter 17. Interesting stuff though. All right, so monocytes and macrophages, again, they are going to be phagocytes that basically are going to be the go-between. They're the scavengers of what the neutrophils left behind. They're gathering the evidence and they're going to bring it to our lymphocytes so that they can interact with it. All right, when we are phagocytosing, we have our pattern recognition receptors on our surface that recognizes the PAM. But the whole reason we got called there was because of chemokines. Again, toe got injured, cells at that injury site started calling for help. Cells nearby them saying, hey, somebody needs help out here. You follow the signal out of the bloodstream to that injury site. That's chemotaxis. We're responding to chemicals, cytokines, and taxiing, moving in response to those chemicals. That's chemotaxis, okay? Once we get there, Our pattern recognition receptors recognize our PAMPs and trigger ingestion. We envelop our whole cell around that pathogen through phagocytosis. The first vesicle, after we eat it up into the vesicle, that vesicle is called a phagosome. I know it says phagolysosome up there. We haven't made the phagolysosome yet. We've made the phagosome. Phagosome, which merges with a lysosome that had digestive and killing enzymes in it. That creates the phagolysosome when we merge them. Then we kill the organism through destruction, and then we excrete the waste. That's how phagocytosis works. Those are the steps of it. Again, we recognize the organisms and then trigger ingestion based on the PAMPs. Those pattern recognition receptors recognize PAMPs. The three major PAMPs were peptidoglycan, LPS, and double-stranded RNA, which is found in viruses. Those are major ones. There are other ones, but definitely know those. Okay? They're patterns. It doesn't tell us much. It tells us they're non-self and that it's okay to eat. Okay? The phagosome is what we internalize that thing into. Then the phagosome that has the thing in it merges with the lysosome that has the digestive enzymes to create the phagolysosome. Now we've injected those digestive enzymes in and started breaking down that organism. It usually takes about 30 minutes to fully kill whatever you ingested or fully break down whatever you ingested. Okay. And then those lysosomes, when we have a ton of them like that, those are what are staining as granules in our granulocytes. Okay. Typically, if we have a lot of them. Now, what is a phagosome? Make sure you read your choices. A phagosome. We had phagocytosis. We had phagocytes. We have phagosomes. We have phagolysosomes. And we have lysosomes. A phagosome is the vesicle that's created when a phagocyte ingests anything foreign. The vesicle that contains the enzymes and chemicals, that's the lysosome. The merging of the two is the phagolysosome. All right. The enzymes, that would be lysozyme and things like that that are inside of it. It also has reactive oxygen species, acids, and things like that. It's a lot that goes into a lysosome. All right. And the type of cell that ingests pathogens or foreign material is a phagocyte. Get the terms down, right? All right, so inflammation, what is it? So once we send out all these signals for damage, we start with inflammation. We start signaling in for our neutrophils to come in and start eating things up. Well, they don't just come by themselves as just the cells. They come in with all the fluids as well. And this fluids that are moving into the area, basically they can come with increased blood flow. So we're increasing the blood flow to the area, which causes redness to appear when you injure yourself. Right? Even if you hit your elbow and it turns red, that's because the signals are being sent to increase blood flow to the area. If there's damage, we need to increase blood flow because we need to move neutrophils in, and we need to start bringing in clotting factors and all sorts of stuff to repair damage and all stuff like this. So the redness is the increase in the blood flow. The fluid moves out too, as the neutrophils and all the clotting factors and all the good fats and the energy, ATP and everything like that's moving in to help feed this whole healing process. That comes with the fluid. It's all moved by the fluid that's moving out of your vessels into the area. That leads to swelling. That's tumor. Okay, so rubor is the redness from the increased blood flow. Tumor is the swelling from the increased fluid into the area. We increase blood flow. Blood is warm, right? Yeah, and if it came from the center of your body, it might be warmer than the rest of the area in your extremities. The warmth is calor, okay? If you've heard of a calorie, that's the heat, basically that's released whenever you have it. It's like a gram of water and turn it into steam. The amount of heat involved in that is called a calorie. And that the amount of energy in terms of heat that a gram of food can release determines how many calories are in a food. Most of the calories that you see on the back of your package, by the way, is kilocalories, not 1 calorie, it's kilocalories. They just abbreviate it because we're simple people. Yes.
Cases like they're I'm not quite sure if I'm pronouncing this right, but dermatographia. Is it like the body's trying to repair itself, or is it just like I don't get it because it turns red like when you burn yourself? Sounds really weird.
Yeah. No. Like this, and then you get the redness there. Yes. So that's instigating that inflammatory response. Your body sees that as the cells are being disturbed. We're going to send blood flow to that area to like basically start the healing process. Now, you might see it as I'm not injured, but at the microscopic level, you are. And so your body is responding to that. Even at the microscopic level, your cells can call out for help, even just a little bit, even just scraping off one little cell or two. Your body sees that as injury and is prepared to start repairing it. So we'll even have blood flow to that. So that is actually triggered by the same response. And in some people, that can be a little bit more reactive than in other people. Yeah, so it just depends on your body and how it responds to that stuff. All right, so it's the same idea. Inflammation is inflammation, regardless of how big the injury is, right? And so this is all that. So rubor, redness from the blood flow to the area. Calor, the warmth from the increasing of the blood flow to the area. The tumors, the swelling of the fluids moving out with the cells and all the reparation and everything that's going to happen to repair the tissues. And dolor is the pain response. Pain response is significantly more important than we realize in alerting us that we have something going wrong with our body. And you know this because if you've seen or heard about this, I made a movie about the guy Novocaine. It's not a joke. People can actually have issues where they can't feel pain. And you might think, oh, that'd be great. It is not. They have a significantly less likely to survive life because they don't know when they're being injured. It's like you cook something on the stove and you place your hand on that hot burner and you're just sitting here talking with somebody with your hand on a hot burner and you have no idea that your hand is literally burning. They can't feel the pain. Can they feel the heat? Technically, maybe, but even that can be stumped. That can be blunted in their response, in their receptors. Because part of reacting to the pain, they can't feel that. So warmth just feels like warmth, but the pain associated with it tells you to remove your hand. They're not getting any signals from any pain whatsoever. Not if they hit their elbow, not if they have their hand on a burner, not if they get stabbed in the gut, not if they have internal injury or anything, no pain whatsoever. So warm, significantly more likely to die at an early age if you can't feel pain. Pain tells you that something is going wrong in your body. So that lets you know. Sometimes that's good, sometimes it's useless. When you work out and you've got sore muscles, you're like, thanks, body. But I'm glad you're getting them. It lets you know that your body's going to heal and make new muscles, great, but it's also not really useful. But generally speaking, it's there for the useful moments. Okay, so that's dolor, that's pain. If you have enough inflammation, you can also have loss of function sometimes. So that can be another sign of inflammatory response. Chronic inflammation, if we have constant influx of neutrophils and things like this to eat up damaged stuff and then causing inflammation, you do lose healthy cells in the process. So if you have chronic inflammation, this can lead to damage in areas that you don't need damage to be happening as a result. So all of that can cause an issue as well, which we know about with autoimmune diseases. Long COVID is associated with overreaction of your inflammatory response. Okay, so that leads to long-term inflammatory response as a result of just having had COVID. So that's what long COVID is. All right, so what's the point of inflammation? The whole point is to, number one, the heat basically helps with the action of the cells. The higher the heat, the more active you get your cells to be in the area. So that's the whole point of increasing the heat, the influx of the fluid and the cells into the area to mobilize to the side to help with the healing process. And we get the whole action of our white blood cells and the signaling molecules and complement and things like this that destroy the microbes and block their function invasion. All right, so here's a question about inflammation. What are the helpful functions of inflammation? We haven't even started chapter 16 yet. Today's lab is going to be a shorter lab, so it is going to be one of those days where we're going to bleed into the lab time a little bit, but we really got to get chapter 16 covered. Luckily, chapter 17 and 18 shouldn't be that big, but today's lab will be done early, so we're going to eat a little bit into lab time today. So this is all of the above one. This is one of those. Yeah. So these are all going to be beneficial functions of the inflammatory response. All right, so basically just to summarize what happens, you step on a nail, bacteria get in, cells that are damaged in the area, including mast cells that are set up in all of our tissues and our extremities, can send out signals, cytokines, saying there's damage in the area. Then that signal causes vascular reactions, increased blood flow into the area, decreased blood flow out of the area, increased buildup of fluid in the area from the vasculature, as well as now we're calling the neutrophils in. Neutrophils get called in to eat up the bacteria. We have an image of some neutrophils eating up bacteria here. And we have the fluid, the neutrophils, and the bacteria, and all the waste of breaking down these guys. That's what pus is. In fact, pus is primarily neutrophils, not bacteria. It's actually primarily neutrophils, primarily your white blood cells. But that's what pus is. It's whenever neutrophils are called into an area to eat up bacteria. And it's all the waste of breaking them down and all the fluid in that area as well. Edema is what we call swelling with the fluid as a result of this whole process. All right. And then after that, we're eating up these bad guys. We're breaking them down. The neutrophils start dying and killing off the bad guys. Macrophages move into the area. They start eating up the neutrophils and the waste they left behind. And then they will call in for our B and T cells to start acting. So again, early changes will be in vasculature and the movement of the white blood cells into the area, including movement of the fluid. So the exudate is the fluid that came from your blood into the tissue. So that's called exudate. Okay, so it has the white blood cells, it has plasma proteins, it has the globulins, the albumin, fibrinogen for blood clotting, and whatever else. So it's all that fluid in the extracellular space. It's what causes the swelling. What you were saying with your dog, Kristen, that fluid, that's exudate from the bloodstream into an area in response to injury or something like that. And that leads to edema. Edema is the swelling from the exudate buildup. And so if you're not moving around, or like if you had breast cancer and we had to remove lymph nodes, and now we have an issue with scarring in the lymphatic tissue and we can't drain fluid from an area properly, we'll have buildup of fluid in the area. Anybody here ever heard of elephantiasis? Maybe even seen it, where you'll have like people will have large swelling of like their whole leg will be like giant, almost like an elephant's leg. And that's usually because parasites build up in the lymphatic system and block the flow of lymph back into the heart. And so the fluid keeps moving out of the vasculature into the area, but can't drain back in. And so there's ways to treat that, but it's actually pretty difficult once it starts to reverse that process. Yeah, you're good.
Not like venous reflux.
I don't know enough about that to answer that. Maybe, probably, I don't know though, yeah. All right, so let's say if I stub my toe and if I've got swelling going on, and I have damage to the tissue in the area, even if like you pinch yourself a little bit, right? You might have damage to the tissue in the area, but maybe not the vasculature. How do the white blood cells get to the area if there's not like an opening in the cells? Well, they actually squeeze between the cells of your capillaries. So while your capillary looks like one little, like tiny little hose, it's actually cells that are connected to each other. And your white blood cells can actually squeeze between those cells. And the process of that is called diapedesis. Diapedesis is the term for when our white blood cells squeeze between our capillary cells to move into the the tissue outside. Okay. All right, there's a question about it, I'm pretty sure. Yeah. All right, what is the term for the mechanism that enables white blood cells to migrate through vessel walls and into surrounding tissue spaces?
Did they squeeze through what?
Between the actual cells of the blood vessels. And so someone asked in one of my other classes about the blood-brain barrier. Okay, so is that a physical method of preventing things from getting into the brain? Yes. So the junctures between the cells and the blood-brain barrier are more tightly regulated. So in other areas of the body, white blood cells can squeeze in and out as they need to. But in the blood-brain barrier, even your white blood cells can't squeeze out. Like it's very much more tightly regulated. We can't let cells that kill self cells into your central nervous system. And that makes sense. When I tell you, if I sever your spinal cord from your brain, can you heal that injury? Well, number one, it will kill you. But even if you had partial severing, can that ever heal? No. So if your cells start going into your brain and start killing infected self cells in your brain, can you have viral infection in your brain cells? Absolutely. It's called encephalitis. That's what encephalitis is. Viruses infecting your actual brain cells. So we can't have T cells, cytotoxic T cells going in there and killing those cells. Absolutely not, because you would lose your brain tissue and die. Can that happen? Yes, but the blood-brain barrier tries to keep that under control as much as possible. In fact, your brain tissue has its own kind of immune function. It's not separate, but it's more downplayed. It's less significant, not as much inflammation, less likely to have damage, but it's not perfectly contained, okay? So we actually have a privileged immune site in our central nervous system, which we'll learn more about in Unit 4. But we have to regulate that inflammation in the brain tissue because it can't cover. So there's benefits to edema and chemotaxis. We're calling in our blood cells. We're calling in all the fluids associated with that and causing cells to move into the area to start healing and things like that. It also basically waters down any toxic substances that could be produced by the bacteria, literally like dilutes them with the fluid that's moving in there. The clots can trap microbes into place so that they can't move out of that area and stuff like that. And then neutrophils are brought in to destroy the microbes. Once inflammation has been going on for a while, we'll call in our macrophages, which they will call in our T cells and our B cells. Then we can have our actual adaptive immune response and clear out the infection a little bit more specifically. Then the tissues can repair and they often leave a scar. Part of our inflammatory process with the calor part of inflammation, the heat, that can be localized, but it can also be systemic if needed. When we have systemic raise of temperature, that's a fever. So there's different levels of fever, low grade, moderate, or high grade fevers. Basically, you don't have a fever unless it's at least 100 degrees, and your doctor might have a more specific thing. My doctor, when I go to like urgent care, I say my doctor, I really only go to urgent care. But you go to urgent care, they're generally like, if it's not 100.5 at least, they don't care. So if you have a 99.5, they're like, cool, no fever, they don't care. It's normal to have your body raised to like 99 and stuff like that. You go for a run outside, a general run. I'm not saying a whole marathon. I'm saying you go for like a 20-minute jog outside. And you come and take your body temperature. What do you think it's going to be? It's going to be 98.6. Probably going to be 99, 99.5, something like that. If it's much higher than that, then you might start getting worried just from physical activity. So your body's temperature can fluctuate. In fact, if you're older, we're more likely to see a lower body temperature and stuff like that. So it just depends. So this is not a hard and fast rule. When you guys get into the nursing program, you'll learn about how geriatric patients have a much lower basal body temperature than otherwise healthy adults.
You know, I wanted to talk about, you just brought up geriatric, but like, why is that a thing that's like, when you get older, like you're like, there's a lot of people in the geriatric.
So up until about 1800s, I think it was around about 1800. People didn't live past 30. You're not supposed to. Your body's not built for that.
No.
So past 30 years old to 1800, people didn't live past 30. Now, when we started introducing hygiene and stuff like that in the 1800s, people lived to 35. I'm not kidding. Once we started with vaccination and proper hygiene with sterilization, not just disinfection, but sterilization in ERs and vaccination and development of antibiotics and things like that. Modern medicine, people started living longer. And that's why the average age now is between 75 to 80, depending on what the census would say. Right? In the US.
Like cancer cells are already in everyone's bodies and it just depends.
Technically, so your cells, number one, they're not meant to live that long. Your body is not meant to replicate your cells for that long. It can't. We talked about this with genetics, right? We talked about this with telomeres. Your cells can only divide so many times, and then they start messing up because you're losing genetic information if you start dividing your cells too much. The longer you live, the more your cells are dividing. Your cells are not immortal. You can't make your cells live longer. That's literally impossible. So your cells have to keep dividing to replace themselves. That is not possible to sustain indefinitely. So if you have to keep replacing the cells, they have to keep multiplying and dividing. You lose genetic material with every time that you replace a cell. So the older you get, that's going to happen. The older you get, mutations that you're building up from sun exposure, mutations you're building up from the food that you eat, mutations that you're building up from the air that you breathe, and whatever else, that your body can't repair them. It's more likely that they're going to slip through the cracks. And so you're more likely to see cancer from that. And it takes a long time sometimes for your body to actually show the cancer that you might be building up, like you're saying. Now, it's not like you necessarily started with the cancer cells from birth, but you might have had sun exposure like me, like I said previously, the millennial era. Many of us sunned in the sun to tan. We did not wear sunscreen. We actually intentionally didn't because we wanted to tan. And so the likelihood that I'm going to have melanoma later on in my life is very high because of that. I wear sunscreen now, but that doesn't help the fact that I might have a cell somewhere in my body that's been multiplying with a mutation that's just been waiting to get out of control somehow. It's multiple factors that lead into that, but the older you get, the more those factors compile. Yes.
Okay, so you know like the, I don't know what it's called, but the actinic flora.
I don't know much about it. Yeah.
I was gonna ask if that to do with like how our immune systems are slowing down and the inflammation's not being.
I don't know much about that one. So is it sort of like, I don't know much about it. I'm just guessing here. Is it sort of like eczema, like related to that, or?
Well, it's like, you know, when people get really old and they like bruise super easily and it's like dark and huge for a very long
Yes.
time.
So
So
that,
looking at that
and
and
like when we
is
get
it-
older, we see more of like what we call spider veins and things like that, like weaker capillaries and you know connections to try to keep tissues supplied with blood as a result of having to adapt to the changes in your body with age. And I feel like that has a lot to do with that. I don't know much about that stuff. There is a whole science between geriatric medicine. Like there's a whole field of geriatric medicine that studies this type of stuff that I'm not very well versed on, but does the immune system play a part in that? Absolutely. Yeah.
Blood thinner or something like that.
Yes. And so that would have to do with like the clotting factors and stuff like that. So the likelihood that we're, you know, and there's other stuff that we're not talking about that can relate to that stuff too, which is like buildup of cholesterol in the blood vessels and all of that stuff. And that's not immune, that's, you know, physiology type stuff. But does that also maybe trigger immune response and stuff like that? Yes. So is there inflammation associated with that? Absolutely. So this is not like learning about the immune response is this box. And then learning about, you know, heart disease is this box. It's all going to be related to one another. So if you have, for example, cholesterol buildup in your coronary arteries, and now we have death of heart muscle as a result of not getting enough blood flow. And now we have to send in the inflammatory response to start to try to repair that issue. Now we've got inflammation in the heart and stuff like that. So it's all always related. Yes.
I think it has something to do a little bit with like their muscle tone. Older people try to have a lower temperature.
Mhm.
Like a lower temperature. Do you think it prevents them from fighting infection as well? Like, can they get a fever as efficiently or not?
Yeah, so it all compounds on itself, right? And so it all does feed back on itself. It's all like just multi-system all the time, all of that stuff. Now, if you talked about, you know, older folks that lift weights still, and they're more mobile, and we know that, and their skeletal structure is more supported, and they're less likely to break a hip and all that too, but they're also going to have stronger immune flow. They're going to have more lymphatic movement and all that stuff too. So they're less prone to like lift weights. Encourage your older folks to lift weights. It's the number one predictor of survivability, basically. You know, beyond not smoking and stuff like that. All right, so pyrogen is any molecule that can trigger fever. They're called pyrogens. Pyro, fire. Think it like, yes, that is right. So a pyrogen increases your body temperature. Signals to increase fever come from inside or outside the body. So if you have a whole lot going on inside the body and nothing from outside, like bacteria or viruses, are triggering a fever, your body can say, we need to have a fever, and produce certain signaling molecules to say fever. Those are endogenous. So certain types of interleukin, like interleukin-1 and tumor necrosis factor, you know, I'm not going to test you on the names of those cytokines, but they are cytokines that can signal to increase fever. However, things like endotoxin and things like certain parts of bacteria and viruses can cause your body to just automatically make a fever too. If it came from outside the body, exogenous. It came from inside the body signal, that's endogenous. Okay. So here's the question. Which of the following would be considered exogenous pyrogens? It's only one of them. Yeah, it's okay. This is why I don't face this on the grade. If someone's kick you out or get frozen or whatever. All right. Good. This will be endotoxin. So tumor necrosis factor, interferon, and interleukin, they're all cytokines made by our body. They're endogenous. Exotoxin, endotoxin or exotoxins, honestly, it wouldn't matter. But toxins made by the bacteria, those are exogenous. They didn't come from our body to signal, but they can be pyrogens. What's the benefit of fever? It increases our body's metabolism, increases our cells to respond. It helps our lymphocytes find the site of the infection. It helps increase the growth of our cells and the multiplication of our cells, but it also inhibits the microbes from growing. The high temperature is unfavorable to many microbes. Obviously, not all of them. Plenty of microbes that can survive a fever. But you get a fever from an opportunistic pathogen that's not built to survive that. Fevers can actually help manage the infection. So it impedes them from being able to multiply, inhibits their enzymes, and it also helps hoard away their nutrients, including iron. So when you have a fever response, it helps hoard away iron so it's not free in the blood for the microbes to use it, because microbes need it too. So decreases the availability of iron, it slows their enzymes and inhibits their growth, but it increases your metabolism, helps your white blood cells find their way to the area and all sorts of stuff. So having said that, here's a question about it. What are some benefits of fever? It's already 10:30. That's okay. The lab should be quick. My other labs got out early, even with this carryover, so we should be okay. All right. So, yes, so good. So it's all the above one again. All the above. All right. So should you treat the fever? It depends on the patient and it depends on the situation and all sorts of that. But generally speaking, it's there to benefit you. So if you are in a moderate to low grade fever, it depends on how uncomfortable you are, really. Okay. Ideally, I would say let the fever run its course. But if you have any other sort of comorbidity going on in your life, you need to address that according with what your doctor says. And if you're a kid, always consult the doctor because they would know better as to whether or not it's appropriate to treat the fever. What is the safest drug to treat a fever? Yeah. The safest drug to treat a fever is acetaminophen, trade name Tylenol, also known as paracetamol. It's all the same thing. So that's the safest thing to treat a fever. Next thing you might reach for would be an NSAID, but NSAIDs can be tough on your stomach. So if the patient, your kid, you or whatever isn't eating a lot of food, don't take an NSAID. Always take NSAIDs with food because they can cause severe stomach damage if you didn't know that, FYI. Okay. Talk to your doctor about it. All right, interferon. Interferon interferes with viral infections. Okay. So if I have a viral infection as a cell, and there's a virus growing inside of me, I can call the neighbor cells with interferon and send out interferon as a signal to my neighbor cells and say, hey, I've got an infection inside of me. You might want to get ready and make whatever you can to defend yourself. So it's a way of signaling the neighbor cells to basically get ready for the impending doom, okay? Cells can make antiviral things and get ready to degrade nucleic acids and things like that. But they don't want to do that all the time because they need their own components. So they can't focus on that. But if they get the signal from interferon, then they'll switch over to being antiviral. Interferon is really useful for fighting viral infections and cancer. So we can actually use interferon as a chemotherapeutic drug. It's usually a very common chemotherapy protocol type of drug. All right, yes, question.
What is interferon? Is it something that your body makes?
It's a cytokine. So yes, so your cell, it is a protein that the cell makes as a signaling molecule, but it's one of the cytokines. Yes, so.
Your cytokine interferon in chemotherapy?
Yes. Well, it could be
I didn't know this.
interferon, so is just a protein that anybody's cells can make. So your interferon is the same as my interferon. That makes sense. Yes. So your cells make interferon, my cells make interferon, all of our cells make interferon. And so we can actually engineer bacteria to make interferon. And then we can use it as a drug as part of our chemotherapy drug targets.
Do they use like the CRISPR-Cas9 anymore?
We don't need to use CRISPR for that. We can actually just use those restriction enzyme techniques to like put interferon gene, remember cDNA, put it into a plasma, and then put that into a bacteria and have bacteria crank out interferon for us. Yeah.
So they're just communicative proteins.
Mm-hmm. Yeah, they're just communication proteins, exactly.
Acids to kill the bacteria or
So it doesn't, but it signals the cell to make those things to group. There's other proteins that it'll make. We won't talk about what they are, but yeah. So there's other proteins that the cell would make. When it gets the interferon signal, it says make proteins to degrade DNA, make antiviral components and stuff like that.
So it's similar like a B cell. It brings things to the fight, not necessarily.
Yes, it's not doing the fighting. Yes, it signals the cell to do the fighting. Yes, exactly. All right, so next is complement. So pretty much all of chapter 16, which again, we have not started even yet. Pretty much all of chapter 16 is talking about how cells direct fighting. And we even talked previously about how neutrophils are eating things up. Macrophages are eating things up, and they're sending signal molecules to induce fever or inflammation and whatever. Complement is a different thing. Complement is proteins that are in your bloodstream right now, 50 of them. They're always there. They're always present in your bloodstream, just waiting to be triggered. Think of them as little mini bombs, and they're just waiting for the thing that lights them. And there's 3 different ways you can light them. We're going to talk about that too. But once they get lit, one gets lit and it lights the other ones. It's a cascade effect. So these 50 different types of proteins, different things can trigger different types of them, but they're always existing in your blood constantly and can be activated by any of these 3 major things. And basically, once they trigger that cascade event, they have different roles that they can fulfill. Complement cascade can lead to helping phagocytes eat things up through a process called opsonization. They can lead to signaling the inflammation response more. And they can lead to creation of the membrane attack complex, which is basically poking holes in the cell. So these are just proteins that do this, not cells getting triggered to make proteins, not cells doing signaling, none of that. These are just proteins that exist in your body that have these functions. The 3 ways you can trigger that the complement cascade: light the bomb that triggers the cascade. You can either light the bomb with the classical complement pathway, which is triggered by antibody. You can light the bomb with lectin binding to a bacterial sugar called mannose. We don't see mannose on any of our cells. So lectin is a protein found in our bloodstream that if it binds mannose, that can trigger lighting the bombs of the complement cascade. And then there's the alternative pathway, basically recognition of PAMPs, general recognition of PAMPs. So if complement sees a PAMP, then it will trigger the alternative pathway. Most commonly LPS that triggers this. Okay, so it's not a cell responding to a PAMP with a pattern recognition receptor. This is proteins reacting to a PAMP. Okay, are we good? Those are the 3 ways. There's a question about it, so let's see if you paid attention. Which complement pathway is triggered by antibody? We're almost done with this chapter. This complement's the last thing, and then we're going to jump right into 16. Huh? Mm-hmm. They all kind of work together, and you like trigger one and it causes it to trigger another, and then that triggers another, and so on and so forth. Yes. Okay, so that's the classical pathway. The classical complement pathway is triggered by antibody. The lectin pathway is triggered by lectin binding to mannose sugar on bacteria. Mouthful one, but it is. And the alternative pathway is triggered by things like LPS. Oops. All right. Basically, you initiate the cascade because you have the presence of the antibody or the PAMP or the lectin binding to mannose. Then that causes the amplification. You lit a bomb, it explodes, causing more bombs to explode, causing more bombs to explode. That's the cascade and amplification. Then polymerization. Once we light the bombs, the parts of the bombs can come together and polymerize, and they work together to do their jobs. Again, opsonization, which is helping create molecular handles to grab onto as phagocytes. Remember how phagocytes have a hard time grabbing onto the sticky capsules? Well, things like that can be overcome by adding molecular handles. That's opsonization. Okay, so they can do that. They can signal inflammation. Once we start polymerizing, they can also form the membrane attack complex, which is a hole that will build with parts of the bombs and literally build a hole in the membrane of our target pathogens. And then they leak their contents out and die because you can't have holes in your cells. Like there's a reason why we have membranes, remember, for osmosis and things like that. We're ruining that, and that'll kill them. So this is something your body can do without relying on the cells. This is something you can do immediately. And obviously, antibody can help that. Yes?
Which step was the opsonization?
That will be part of the, it's a separate thing than the membrane attack complex. So I would say it's related to the polymerization step, but it's not necessarily have to fall in one of those steps. But yeah, it's related to that. So there is a video for that, but we're not going to watch it. If you want to watch it, it explains the complement cascade. It has a little video that like shows it happening. We just don't have the time for it. Yes. So chapter 16 now, we have a lot to cover still, so keep in mind. Now we've introduced a lot of the concepts behind chapter 16, so it shouldn't be too long. Here we go. make sure we're signed in real quick. Like I said, we're gonna leak over into lab. It's already 10:38. But it shouldn't take us long. Oh yeah, we'll be fine.
What about pediatric leukemia?
Yes, so that's usually going to be related to genetic predisposition. There's some sort of genetic marker that makes it more likely that your cells are going to overgrow. And then it's usually like a type of cell. And so like a cell that sees Barry's eyeball. Okay, that cell has some sort of defect in its genetics. And then we happen to activate that cell with a pathogen or something else. And then it got triggered to multiply, and then it just went out of control.
Usually pretty treatable. I mean,
It can be if caught early enough. It just depends. And so it depends on the type of leukemia, what type of cell got activated. So if it is lymphoma, then we might be able to just target, that cell line, or even like get rid of your bone marrow. So remember, all of your white blood cells come from your bone marrow. I can irradiate you. All of those stem cells are very susceptible to being killed by radiation. So I can irradiate you, eliminate all of your white blood cells, and replace it with somebody else's white blood cells. And now you have new white blood cells, and none of them have cancer. So we can do that if the cancer hasn't spread too far in your body and stuff like that. Yes.
Those send out cytokines.
Yes. Yeah. All of our white blood cells can. Even your tissue, like your normal tissue, can send out like cytokines. Just think of a cytokine as just like any signaling molecule between cells. Yeah. All right. Okay. Cool. All right. So we're going to keep going. Chapter 16. Here's our learning objectives. Again, this is all dealing with that third line of immunity. Okay. So this is usually a result of our B and our T cells, and it relies on immunocompetence, and a very specific immunocompetence. Immunocompetence refers to our ability to react with specific individual foreign molecules, foreign substances. It's, again, specific to an antigen. So we're not talking about general recognition of patterns, not seeing that there is endotoxin, but now seeing Barry's eyeball, like a specific antigen, okay? And so that's specificity, and these are what lead to memory. You don't get memory from your neutrophil recognizing a PAMP. You don't get memory from a macrophage recognizing a PAMP. You get memory from your B or your T cell reacting to, somebody's, say somebody's eye. I'm always picking on Barry here because it's just right here, but Barry's eyeball. Once we've seen Barry's eyeball, we remember Barry's eyeball. And so that's the idea there.
Brain too.
Yeah. All right. So antigens, also known as immunogens. This is getting into those specific antigens now, not PAMPs anymore. Now very specific antigens. These are like Barry's eyeball. Okay? It's the part of the pathogen that we react to. I always forget to bring my things, but I'll just show you an example of it. So if this is the pathogen, this is my keychain. It's a lot going on with it. I'm aware of it. All right. But this is my keychain. All right. This is my key fob. This might be an antigen. So it's part of the pathogen. Okay, the keychain is the pathogen. The key fob might be an antigen on it. Or like whatever this is, the little two voodoo doll guys. Okay, so we'll do the voodoo doll guys here. Might be an antigen. Okay. Part of the pathogen. All right. Because we're getting somewhere. Antigens or immunogens can be any type of a molecule, any type. It could be proteins, polysaccharides, all sorts of stuff. More commonly, it's going to be protein or polysaccharide than it is like lipids or something else, okay? Bigger molecules, it's easier to react to them. Smaller molecules, less easy. And more complex, the better they're going to be as an antigen. We need to be able to recognize them as foreign or self. And the more detail that they have in them, the more we can recognize it as foreign. Okay. Again, if I look at my key fob, there's not a lot of detail. It's nice and smooth. It might be less good of an antigen than this, which has a lot of detail going on. We can pick it out as specific. Anybody might have a key fob. Well, this, not all y'all got this, whatever it is, okay? I don't know, my husband got it for me. Little voodoo lovers, okay? That's what they are. All right, good antigens, we want them complex. The bigger, the better. Typically protein and polysaccharide or some combination of protein and polysaccharide. All right. The part of the antigen that our lymphocyte is going to interact with is called the epitope. So while this whole thing might be the antigen, like our viral spike, okay? If this is my virus's spike, this little voodoo doll thing, right? But the antibody interacts with this leg here on it. So this whole thing might be the antigen, the viral spike, but the antibody recognizes this leg. That's the epitope. Okay? Does that answer your question, Brandon? Okay. All right, so that's the epitope. It's the part of the antigen that we actually interact with. Do we interact with the whole thing? Not typically. It's typically part of the antigen. So viral spike, which part of the virus spike? The little protruding part, whatever. All right, so that's the epitope. All right, a different term that relates to this is a hapten. Haptens are really tiny, small things. So if I were to say that this eraser, if the eraser comes out, watch be one of those, I can't get it. Okay. If this eraser is the part that I want to target, let's say it's the tetanus toxin, and I want to create an antibody against the tetanus toxin. Well, for me to initiate a reaction with it, it might be too small. Tetanus toxin is too small, by the way. It's too small. Our body can't interact with the tetanus toxin. That's why if I were to just inject you with tetanus toxin, you'd just get sick from the toxin. Your body can't create an antibody to it. Instead, we link the tetanus toxin to a carrier. Now there's enough for my antibody to interact with it. And we can create an antibody to that epitope now because we linked the hapten, which was the whole thing that was too small, to a carrier. Does that make sense? Are we following? Image of it, okay? Here, too small for the mouse to react to, doesn't make an antibody, link it to something else. Now it's big enough for us to create a proper response to. There's enough to grab onto, there's enough to create signaling for. Now, with tetanus, we know we have the Tdap vaccine, tetanus, diphtheria, and pertussis. As it turns out, those are all toxins that are too small for us to react to. So we link them together. And now, because there's multiple parts that we can now interact with as a big mass, we can create antibodies to the individual parts. We use each other
So
as
instead of
carriers.
using the carriers, they just link them all together.
Cool stuff, right? All right, so here, what is the term for the part of the antigen that is recognized by lymphocytes? Again, this will be this part here. If this is the whole antigen, the part that we're actually interacting with, what's the term for that? Right, so that'll be the epitope, the epitope. Good job, guys. All right, alloantigens. My cells have markers on their surface that indicate to me that it's me, that it's self. Your cells have markers on them that indicate to you that it is self. Some of those markers are the same between us, and some of them are slightly different just because of genetics and things like that. All right, those are called alloantigens. An example of that is that my red blood cells, I have A positive blood. So my red blood cells have A antigen on them. If your red blood cells have B antigen, my body sees that as foreign. It's never seen B, and it doesn't want to see B, and it's ready to attack B. That's foreign. Is that actually really foreign or dangerous? No, but my body can't tell the difference. So those are alloantigens. So antigens that are associated with other people just because they're different. They have different markers, okay? We'll talk about specific alloantigens in chapter 17 next time and how like we predict for that with blood type rejection and things like that. So this often is using, that information is responsible for blood type differences. So the ABO group, the RH group, which is whether you're positive or negative, is a different type of group. Okay, so that's a marker. The ABO is a marker. And also MHC, which we're going to talk about in this chapter, is another marker. Typically MHC1. MHC is also known as major histocompatibility complex. And it can be essentially different from person to person. And we use it as a predictor as to whether or not you can receive a donation of tissue from person to person, is the MHC marker. We'll talk about what it does really in the body. It has its own function in the body in a little bit. Some pathogens have super antigens. And so these are things that overstimulate the immune response. And endotoxin is one of them, but other organisms create other types of toxins that basically like trigger the immune response so high, it overreacts and then makes you really, really sick. And we call them super antigens. And toxic shock syndrome is a result of that type of a response. So there's a toxin associated with those bacteria that can cause a super antigen reaction. All right, so some of our cells, like macrophages, remember, macrophages are going out into the field and picking up all of the debris left behind by neutrophils. Neutrophils were just out there killing anything it saw as foreign. And macrophages are going up and eating that stuff. And they're going out there and they're saying, hey, I found this. I found this thing, and holding it up. Because macrophages are antigen presenting cells. They present the antigen to our lymphocytes, to our T cells specifically. So if they picked it up, they saw this lying around outside, they phagocytosed it, and then they are presenting it from outside. That would be MHC2 that they would present it on. If they found it inside of their own cell, that would be MHC1 that they presented it on. Any cell in your entire body that has a nucleus has MHC1, because any cell in your body that has a nucleus can propagate viruses. Any cell in your body that has a nucleus can become cancer. So they all have to be able to call out and say, I found something wrong inside of me. The inside flag that presents antigen, it's very, very general presentation. MHC1 presents things found on the inside. MHC2 presents things found on the outside. I know it feels confusing now, but it's about to get even more confusing. And you must know the difference. Yes,
The outside of one,
the cell.
from the cell.
Yes. So if you're a macrophage and you're chilling, you're going around, you're picking up all the crap that neutrophils left behind, that was outside of your cell, you're eating it up from outside of the cell. Did you bring it inside of your cell? Yes. Outside the cell, yes. So you quite literally, you stub your toe, you got bacteria coming into your body. Those bacteria are not in the cell, they're in your body, but they're not in the cells.
So it's
They're
not the
outside
cell.
of the cells.
It's not the cell, but the non-cell.
It is the cell and the non-cell.
Okay.
Okay, so walk through it from the beginning. I stub my toe, the bacteria get into the body. Cells nearby, because there's damage, start sending out cytokines asking for help, right? There's help, help, there's damage, there's damage. They don't know that there's bacteria. They can't know that there's bacteria. They don't know yet. They start calling for help. Neutrophils come in from the bloodstream. They recognize PAMPs. And when so they follow chemotaxis from the cytokines. They recognize the PAMPs with their pattern recognition receptors, and that let them say, this is foreign, I'm now going to eat this up. They ate it up, they broke it down. So they had to recognize that it was non-self. General recognition of non-self there. I know it's not self, I ate it up and digested it. They eat it up and digest it and either die or poop out the waste. So now there's all this waste left behind. Macrophages now come in and they pick up the waste. Again, phagocytosis. They come in and they say, PAMP, non-self, eat it up. So it's a second level of eating it. But they're picking it up as evidence, not to break it down and kill it necessarily. They can do that too. But they're really there to pick up the evidence of what the neutrophils left behind. And they're going to say, I picked this up outside of my cell. So therefore I put this on an MHC2 to call for help, to go talk to the T cells, as we'll say, okay? So if I picked it up from outside of the cell, found it outside, then it's going on MHC2. If I found it within my own cell, it goes on MHC1. So if you have a virus infecting you, or you have cancer, something going wrong inside of you, then you'll put it on MHC1. Yes.
So would it have to be literally like another macrophage? That's, I mean, how does it know that it's self? Because the macrophage is a cell, and then it goes and eats up pieces.
Mhm.
And then how does
It
know?
uses a PAMP, again, it's going to be a pattern recognition receptor. on our macrophage that is gonna recognize self versus non-self. The PAMP is what tells it that it's non-self.
No PAM, then it's gonna know it's gonna be.
And it'll come in and right, so I say no PAMP, and I'm telling you guys that PAMPs are gonna be things like LPS and no toxin and stuff, but there's way more things that they can recognize as non-self. But it's not saying this is Barry's eyeball until I eat it up. It's just saying this is not self, so I eat it up.
Yeah.
And it can eat up debris too. So let's say you have damaged cells, it'll eat that up too. So it pretty much comes in and eat up any debris that isn't a healthy living cell. And there's other markers that will indicate that to the cell, to the macrophage. So it basically comes in, uses receptors all over its surface and says, you're a debris, I eat you up now. And if it found it outside of the cell, it will present it on MHC2. Yes.
I would think, I don't know if it's body not part of the body, like you ever heard of it.
Yeah.
Is that part of it? Like, because I've heard people say, oh, your body isn't aware that your eye is a thing. So therefore, if it's aware of it, then it's gonna destroy your eye or something.
Kind of. So that's a very miseducated way of breaking it down. But what it is your eye is also an immune privileged site. So like your brain is. So that's like people being very uneducated about the fact that your brain does the same thing. So if we destroy, so think of it this way. When we were animals living out in the wild and we had to depend on seeing that there were predators or seeing where our food was or seeing where we were going, could you afford to lose your eyesight? Absolutely not, right? Same level of you can't afford to lose your brain. Like you literally, if you were blind back in caveman days, you were screwed. There was no surviving it, right? And so your eye is an immune privileged site too. So we can't have too much inflammation in our eyes because that would cause eye damage. Now your eye can heal. That's the difference. But you can't risk even partial loss of vision. Now we can. We can put you in a hospital and let you recover. But evolutionarily speaking. So yes, so the eye is an immune privileged site, which protects it from our immune system's overreaction. So we have a different type of immune response going on in our eye and in our brain to protect those tissues from being damaged. Does that make sense? So it's just that kind of a thinking. Now, how strict it is as far as your immune system might see your eye as foreign and could attack it, there's a little bit of crossover there. Yes, because of the training in our body. Like we didn't need to tell our body that our eye was self because it was an immune privileged site where those cells shouldn't be going anyway. That makes sense. But it's a lot more complicated than that.
Your body could do that to like any organ.
Yes, it could do that to any organ, right? And it can, and kind of does do that with any organ, but just we can risk to have damage done to the other organs. We can't risk to have it done to the eye or the brain. And so they are immune privileged sites.
Well, I actually like this. I feel like the eye was covered by like some kind of.
So completely like,
So like,
yeah.
like the cells can get so like it's covered or something.
yeah, it just has to deal with like the training, and it has to do with, it's sort of like the blood-brain barrier. It just selects what cells can get in and out of there, and controls that inflammatory response. So it is a little bit physical, but it's not as like obviously physical as that. All right, good questions. We're going to talk more about MHC1 and MHC2. So if we're a little bit lost on MHC1 and MHC2, we're about to get into it a little bit more. Okay, so hang in there. We're gonna have a little question about it first. Let's see what we can get out of it. I don't remember this. Oh yeah, which of the following choices, which of these cells can act as antigen presenting cells? I even skipped over it a little bit. So, this is one that you definitely need to know as well. We have our phagocytes and we have our antigen presenting cells. Neutrophils can go into a site and eat up things based on PAMPs, but they'll just eat it up and poop it out. They can't present the antigen. Macrophages, which come from monocytes and macrophages are essentially kind of the same thing, okay? But once the monocyte moves into the tissue, it becomes a macrophage, so we focus on it a little bit more. Monocytes, macrophages, and dendritic cells, essentially all the same thing. They can be antigen-presenting cells. B cells can too. B cells are not phagocytes. So if a B cell can be an antigen-presenting cell, and an antigen-presenting cell is anything that can pick up something outside and present it on MHC2 and trigger the response for outside stuff. How did the B cell get the antigen, take it in, and put it on MHC2? It uses the B cell receptor. So instead of recognizing a PAMP, The B cell receptor recognizes a specific antigen. So B cells have immunoglobulins, which are like membrane-bound antibodies. They have a membrane-bound version of an antibody on their outside. And that antibody on their outside recognizes Barry's eyeball, and only Barry's eyeball. Nobody else's eyeball. No LPS, no endotoxin, no peptidoglycan or anything general like that. Its marker only recognizes that specific antigen. So that one B cell that only recognizes Barry's eyeball finally sees Barry's eyeball on its little membrane-bound antibody receptor. Once that binds, and it's like, I've been waiting on Barry's eyeball for so long, I got the eyeball. It takes it inside and then wraps it up onto MHC2. And it says, Mr. T helper cell, which is going to be our captain, Mr. T helper cell, I found Barry's eyeball. And it holds it out on MHC2 and says it that way. So instead of relying on PAMPs to just eat up anything and present anything outside on MHC2, these cells pick up only specific antigen and present that on MHC2. Okay. We're going to have pictures and stuff, so hopefully it'll help a little bit. All right. So what is MHC? We have MHC1 and we have MHC2, and they are different things and they do different things. MHC is a molecule found on the surface of our cells that can present antigen to T cells. MHC is major histocompatibility complex. That's what it stands for, okay? It's a molecule on the surface of cells that presents antigen to T cells. We'll just say T cells because it's not just T helper cells, T cells, to all T cells and only T cells, to T cells. So that's MHC. MHC1, okay, whatever, MHC1, we have 1, is found on all cells that have a nucleus. On all cells, and it presents antigen to CD8 cytotoxic T cells. Really want you to focus on that CD8 because MHC1 is recognized by that CD8 molecule. So this is where it's going to start getting more complicated. This is not the end of the complicated. This is the beginning of the complicated. Okay. MHC2, I'll put the number 2. It's usually written with Roman numerals, but I don't care that you know that. I just care more that you know the difference between 1 and 2. Okay. MHC2 is found only on antigen presenting cells. That's going to be your B cells and your monocytes and their derivatives. So B cells or monocytes, macrophages, dendritic cells. They present antigen only to CD4 helper T cells. CD4 helper T cells are the captains. Okay. All right. One more thing about these. MHC1 is found on all cells. It presents antigen, but more specifically, it presents inside antigen. Whereas MHC2 presents outside antigen. Again, if we're a macrophage and we're perusing around what all the neutrophils left behind after eating up the damage, the debris, and the bacteria from stubbing your toe, the neutrophils left behind all their stuff. The macrophages come around and eat up all the waste that was found outside of the macrophage, outside of the macrophage. That macrophage found it outside. It phagocytosed what was outside and brought it in to put it onto MHC2. Are we following that? If you are a lung cell and you're infected inside with the flu virus, you will say, oh my gosh, what's on inside of me, and present the part of the flu virus on MHC1. Any cell that's infected inside can present on MHC1. Only antigen presenting cells can pick up antigen from outside and present it on MHC2. This is important because your captains, your CD4 positive captains, can only be activated when antigen is presented on MHC2. And MHC2 is only found on antigen presenting cells. Antigen presenting cells only present antigen from outside. And those captains regulate all other cell responses in your adaptive response. Are we ready? Because it's about to get real, okay? Okay, so here we go. Virus, outside. Viruses, in order to get inside, had to be outside first, correct? Can we agree on that? Okay. And how do viruses spread from cell to cell? Outside, typically. They don't just migrate from in a cell to another cell. They had to go outside to get in. Okay, so we have outside pathogen. And we have a macrophage eating up that pathogen or that part of a pathogen. This is a macrophage. It found it, again, outside. It takes it in and puts part of that pathogen, the antigen part, on MHC2 because it found it outside. Here's the antigen, actually. Let's say it's the spike protein. Okay. MHC2 is only recognized by what type of a cell? We can look at the thing on the right. CD4 positive T helper cell, the only cell that will see that. So here we go. Here we go. This part of the T cell is called the T cell receptor. T cell receptor. It's like an antibody on the surface of a T cell. It only recognizes that antigen. It's an antigen specific reaction. Here is the cell. This is a T helper. T helpers have what marker on their surface? CD4. Here's how it works. CD4 recognizes, literally interacts with MHC2. So the T cell receptor sees the antigen, but the CD4 recognizes that it's MHC2. Now that T cell receptor could interact with antigen anywhere, anytime, any part of any time of day. But it doesn't matter unless CD4 also recognizes MHC2. Those two things must happen. So we are T cell receptor seeing the antigen. Again, T cell receptor seeing the antigen being presented on MHC2. MHC2 has that antigen. T cell receptor sees it and says, that's Barry's eyeball. CD4 says, that's MHC2. This is correct. We're doing the thing. We're having it. It's happening, guys. MHC2 is presenting this. CD4 says, yep, that's MHC2. And then the T cell receptor says, this is Barry's eyeball. Those things must happen. This is important. We need it. It's a double check, right? We're not just interacting with the antigen. We have a signal saying we're interacting it the way we're supposed to. Because otherwise you're interacting potentially the wrong pathway. You could be interacting with self. You could be interacting with blah, 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 things that shouldn't be happening. This is a check. Okay. So that check happens. CD4 sees MHC2, which is presenting the antigen, which is seen by the T cell receptor. When this happens, it is fireworks for this T cell. When this happens, the T cell gets activated. Now that T cell is able to proliferate, make copies of itself. So it'll make a T helper, it'll make a T helper, it'll make a T helper, it'll make a whole bunch of T helpers. And they're all the same thing at first. And then each of these, at first they're just T helpers, just very general T helpers. Each of these will differentiate. This T helper becomes a T helper memory. This T helper becomes a T helper regulator. This T helper becomes a TH1, and this T helper becomes a TH2. Anytime you activate a T helper, all of these guys are made. This is important because these two, TH1 and TH2, these are effector cells. A T regulator is just there to make sure we don't get out of control. A T memory is just there so that if we see this again, we don't have to go through this whole process again. But the TH1 and the TH2 are effector cells. They're the ones that do the job. TH1 cells give signals, the CD8 cytotoxic response. The CD8 positive T cells can't react. And we'll talk about what they do in a moment, okay? They can't react unless the TH1 captain tells them they can react. Okay? The T helper 2 cells, these signal B cells to make antibody. B cells cannot make antibody without the TH2 guys telling them. So again, we had to see outside antigen to activate our T helper cell. We are able to do that because the CD4 on the helper saw the MHC2. Okay? Now that T helper is activated, now we can get the TH1 and the TH2s. The TH1s give the signal for the inside response. That's their cytotoxic response. The TH2s give the signal for the outside response. That's the antibody stuff. Okay, so that's generally what they're going to be doing. So again, inside response versus outside. But you have to see outside to be able to modulate both. Okay, are we there with the helpers? Yes, question.
Hopefully clarify this right. So it would have to be the THM or THR to become the CD8 or a MHC1.
We haven't even gotten there yet. Yes, okay. So if we activated our T helper, again, this T helper up here in the pink, this is a naive. It is not TH1, it is not TH2, it is not TH regulatory, it is not TH memory. It's just a TH stuff. It got activated. It multiplied to many TH cells. Some of them turned into memory, some of them turned into regulator, some of them turned into TH1, and some of them turned into TH2. The regulators literally just send signals so that we don't overreact. That's all they do. The memories literally just exist to wait to see this again 10 years down the road. That's all they do. The ones that are actually doing the job right now, the TH1 and the TH2s. The TH1s, they send signals that modulate the CD8 responses. The TH2s, they send signals to modulate the B cell response. That's all they do is send signals. They do not do anything else, but they are the captains. B cells cannot make antibodies without the TH2 cells saying they can. Cytotoxic T cells, they're the guys that go out to your lung cell that's infected saying, help, I found something inside of me, some part of something, I don't know what it is, inside, presenting it on MHC1. So yes, so it could be Barry's eyeball, but think of it also as like you're in the movie Alien, okay? And you've got a chest burster inside of you. It's your way of saying like, I have a chest burster inside me, I found this inside of me. And the cytotoxic T cell reacts with MHC1. Remember, any cell can call out for help for something inside of them. If they're calling out from inside stuff, that's presented on MHC1. The only cells that see MHC1 are cytotoxic T cells, not T helper. Cytotoxic T cell. The cytotoxic T cell reacts the exact same way. So we'll draw it out too. So you've got an infected lung cell. We'll start with this. A lung cell that has a virus inside. So it's going to present that on MHC1. Again, it might be the viral spike protein, who knows? And it's the same exact thing. We have a T cell receptor that is antigen specific on the cytotoxic. T cell. But instead of this being CD4, what is this now? CD8. Okay. This will activate but it's a problem. This can't cause activation. Actually, I should have drawn this differently. Okay, there we go. This interaction here will allow activation only if, come on, work with me, pen. I'm trying to draw all the way across here. We get the signal from TH2. Sorry, TH1. Good. I'm glad that I'm already confusing you. TH1. Okay. If we have MHC1 interacting with the CD8 T cell, the T helper that sends the signal for the go-ahead is the TH1. So if it's MHC1, TH1 sends the signal. It doesn't interact with MHC1. It sends the signal that lets that CD8 cell be activated. So this is like a 3-step check now. So think of a TH cell over on the left that we first saw there. Only needed to see the antigen with the T cell receptor and get the CD4 saying we got MHC2. But our cytotoxic T cell here, this guy needs to see the antigen, it needs CD8 to see MHC1, and it needs the signal from the TH1 cell, the activated T helper. It must have all 3 of those things because this guy is not playing around. This guy is going to then make granzymes and perforins. What do you think a perforin does? It perforates. It's going to put holes in the lung cell that then the granzymes then come in and kill the cell. So for sure, we need 3 checkpoints for this guy. He can't just see the antigen and have CD8 see MHC1. That is not enough. We must have a captain saying, you may go now, because we cannot let this get out of hand. Otherwise, he will kill cells that he's not supposed to. You want to see how devastating that is? Ask somebody that has type 1 diabetes, because these are the cells that are killing the insulin-producing cells. Now imagine that this cell could see anybody else's MHC1 and could just kill your cell. That's what happens in organ rejection. And it can happen within hours. We can destroy that organ within hours if this goes unchecked. So that's why it's so important that if I give you my kidney, that your MHC looks like my MHC, because otherwise we'll just see the MHC is foreign and all bets are off. Make sense? Okay, are we tracking this a little bit better? Do you have a question?
So the damaged lung cell,
Mhm.
it brings the MHC1. The MHC1 is on the damaged lung cell.
Yes, all nucleated cells have MHC1. Yes,
And then for the outside pathogen, so the outside, going to the other one.
yes.
The outside pathogen is there, the macrophage comes up, and it has the MHC2 on it to start the chain.
So
Okay.
it's MHC1 that presents inside antigen. MHC2 presents outside, and MHC2 is only found on antigen presenting cells like macrophages, monocytes, dendritic cells, and B cells. Because they're the only ones that can deal with that. They're the only ones built for that.
So it could be any APC.
Yes, any APC. I used a macrophage
Yeah.
as an example. Yes, yes. Okay, are we getting there? Okay. And it's a lot, and you're probably gonna have to rewatch it and like walk yourselves through it and stuff like that. For sure, get it down. What the CD4, what the MHC, what roles they're playing and what they're doing. MHC only presents to T cells, period. Start there. MHC1 presents to the CD8 cytotoxic. MHC2 presents to the T helper CD4. Now, we saw here how important a T helper is, right? You can't have cytotoxic T cells kill infected cells without a T helper. So we saw that there. So you might be wondering, what's the B cell do? So the B cell, basically here's a B cell. And it has its little marker on its surface, which is an immunoglobulin, specifically its B cell receptor, the B cell receptor BCR is IgD. IgD, it's a type of immunoglobulin. There's 5 types of immunoglobulins. All right, so IgD is the surface receptor, and it again sees that same spike protein that was on that virus outside. This then allows it to take that inside. Again, this is a very antigen specific. Recognition, okay? It takes it inside and presents it on MHC2. Here's our spike. Okay. And then our TH2 T cell receptor, again, antigen specific, and CD4, again, recognizes MHC2. This allows this to then activate the B cell. Now, did you notice a thing? Because I'm hoping that you noticed a thing. And that is that this TH2 was what was required here. This TH2. So the TH2 will activate our B cells. The B cell then proliferates and differentiates. Same thing here. We'll make B cells that are memory, B cells that regulate, and then effector B cells. Effector B cells are called plasma cells. They make the antibody. So now our B cell can turn into whatever it needs to do. The B memory, B regulator, and the plasma B cell that makes the secreted antibody. Okay. So TH1 modulates your CD8 cells to be able to kill infected cells. TH2 modulates your B cells to make antibody. If you do not activate your captain, you can't make antibody. If you do not activate your captain, your T helper cell, you cannot have your killing of your infected cells. So now, are we tracking that? HIV infects CD4 positive T cells, all of them. We said it before, we've introduced the concept before. Does it make sense now why that's so devastating? What AIDS is? Acquired immunodeficiency. Because if HIV infects all of your CD4 cells, you cannot activate cytotoxic cells and you cannot make antibody. Can't do it. The chills of it. When you start to see it, when you start to understand it. Anytime your body sees anything foreign, we need to make our captains in order to make antibody or to fight off infections inside of your cells, period. So if you take away those captains, you can't have those responses. That is also why T cell deficiencies are going to be more impactful than B cell deficiencies by far in our patients. We'll talk about that in chapter 17. Okay? All right, are we tracking this? This is our immune response. On my review on Thursday, I'm going to walk through all of this again without talking the slides. You will have learned the stuff. You will have seen the stuff. We're going to talk through the whole response again from innate all the way through all of this. That's why my review is going to be on Thursday. So I'll record it and post it. So if you can't make it, that's fine. But I'm not going to do the slides like I usually do for my reviews, where we like have the content and we have the little questions. I'm not doing that this Thursday. This Thursday is going to be a walkthrough of the immune response again, but you'll have better perspective because you will have learned the stuff first. Okay. All right. So again, I know we are at 11:22. I'm going to go through the slides. We're just going to click through ones that I've already covered the content. Okay. All right, class 1, we talked about presents to our cytotoxic cells. There's a quiz question. What do MHC molecules do? Hopefully, don't read it on my screen. Read it on your screen. Yeah.
No.
Yeah, if you can read my writing, you're welcome to use it as a cheat sheet. But what does MHC1 molecule do? Presents inside antigen to Great job. Presents inside to CD8s. All right, class 2 presents outside to CD4s. Okay, that's probably what the question is. I'm just guessing. I don't know. Multi-select. Which of the following applies to MHC2 molecules? Select all that apply. It is written over here. It's just hard to see it on the screen. All right, it is found only on antigen presenting cells, and it presents outside antigen to T helper cells. Remember, outside antigen is the part you guys got tripped on here. Yes, so you want to be sure it presents it to T helpers. So that's the part. Read your choices. Okay, the B cell receptor or the T cell receptor. T cell receptor and B cell receptor, they do essentially the same thing. It's just the B cell receptor is based on an antibody that's bound to a surface. Otherwise, they're very similar to one another. And they have heavy and light chain, okay? So the heavy chain is that long Y part. The light chain is this little side part. The top part where we bind with our antigen, where you always see me talking about binding with the antigen at that end there, at the Y, the tips of the Ys, okay? That part is the variable region. Those are different on every single antibody and interact with different antigens. That's the variable region. The antigen binding part, variable. That bottom part, the part that's bound to the surface of our B cell, or it's like the base of the Y as an antibody that's floating around, right? That's called the constant region. And it is different based on the function of the antibody, okay? And we'll talk about that in a moment. The T cell receptor is basically set up the exact same way, but it's just a different shape. And it's smaller, but it still has the variability. And it's always bound to the surface, never secreted like antibody is. Always bound to the surface of the T cell. We reassort genes and then add in and remove nucleotides in order to get our variability in our variable region. There is a video on it that I'm not going to play, but it is in the recording that is online. Okay, and it talks about that variability. Like I said previously, the whole idea is with clonal selection, you only want to multiply out the T cell that sees Barry's eyeball. So if I saw Barry's eyeball, I only want to multiply the T cell that sees Barry's eyeball. I don't want to multiply out the one that sees Megan's hair or Professor Eo's foot or anything like that, right? We only want to multiply out that T cell. That's the idea of clonal selection. Clonal deletion is something that happens if I have a T cell that I made that sees anything self, I delete it, I kill that cell. We don't want to propagate that cell, we don't want to risk it. Is that a perfect process? No. But during your development, your body tries to get rid of as many T cells as can see self as it possibly can, or as many B cells that can see self as it possibly can. That's clonal deletion. Clonal selection is multiplying out only the T cell that only sees Barry's eyeball, that sees that antigen. And then we multiply it, we proliferate it to a whole bunch of T cells, and then change them to their different roles. That's the differentiation part. Proliferate, then differentiate. All right, so here's a little image of that. Again, delete any anti-self guys through clonal deletion. Then we have a whole repertoire of billions of different T cells. One of them sees Barry's eyeball. One of them sees Megan's hair. One of them sees Professor Yo's foot. We have billions of them. And that one gets activated and we only multiply it out and give it its different roles. Immune tolerance is tolerating self and only attacking non-self. Once we are making our B and our T cells in our bone marrow, the B cells typically will hang out in the lymph nodes and wait to see antigen there. And that's where they're going to multiply out once they get activated in the lymph nodes. And that's why your lymph nodes will get really swollen and activated and stuff. The T cells typically circulate throughout your circulatory system, throughout the tissues in the lymph. They migrate in the periphery a lot. Once they see their antigen, they'll move to the lymph node to try to interact with B cells and activate them too. But they don't live there. They just only interact there whenever they're activated. Okay. Cell-mediated immunity refers to our T cell actions. So our T helper cells are cells that direct everything. The cytotoxic T cells are cells that kill infected cells. They're doing their jobs as the cells. B cells, though, they direct humoral immunity because they make soluble protein that's secreted, and then the protein does the work. Okay, so that's the difference there. Let me see what this is going to get into. We talked about CD4 and MHC. I'm gonna skip the question, sorry, no offense. Remember you have your Kahoots available on your modules if you ever wanted to play through them. And I will send out a email with all of them linked together too. Before Unit 3 exam. T helper cells, most important ones are the TH1 and the TH2. Those are the ones I really want you to be focusing on, okay? There's a little video about it.
Once T helper CD4 cells come in contact with foreign antigens attached to MHC2
Stereo.
molecules, they become activated. This activation results in the production and release of interleukin-2 cytokines.
Cytokine is
The
signaling
released
molecule.
interleukin-2 cytokines come back in autocrine fashion to activate their releasing CD4 cells. This activation induces a cascade of intracellular reactions, ending with the replication of T helper cells to 2 new T cell lines, T helper 2 CD4 cells and T helper 1 CD4 cells. T helper 2 cells are the main line for antibody immunity, which is called humoral immunity, whereas T helper 1 cells are actively involved in cell-mediated immunity.
There are other videos made by this person if you wanted to look it up. Again, this is found in your lecture recording and stuff on Canvas if you guys wanted to go back and watch the videos for this. But that is the gist of how that's going to work. And then remember, to activate your CD8 cell interacting with MHC1, you need TH1 cells to send the signal. It is interleukin-2 that is the signal that it sends, but I am not going to test you on that because there's a lot of cytokines and they all have different functions. That might be important if you go into any sort of cancer medicine, but it probably won't be outside of that. So when I tell you this is the tip of the iceberg, I mean it's the tip, okay? There's a lot. Once we activate our cytotoxic T cells, like I said, they're activated by MHC1 presenting antigen, and they will produce granzymes and perforins. We're going to skip it. The perforins poke the holes that let the granzymes in, and then that will lead to the cell dying. Okay, they will attack virally infected cells, cancer cells, and cells from other people or animals. They can, it's basically, that's all inside stuff to them. Okay. We'll talk about that more in chapter 17, like how that affects autoimmunity or rejection of organ grafts and things like that. Once we're doing our B cells, again, they recognize everything with their immunoglobulin. That is their surface receptor, or they make antibody, which is a soluble version of that immunoglobulin surface receptor. All right, so we will activate them by interacting with the antigen with that surface receptor and getting the signal for the activation from our TH2 cells. Then we'll make our plasma cells that produce antibody. These snowflakes are the antibody. I know it looks like snowflakes, but that's a type of antibody that we make first. There are 5 different types of antibodies that have 5 different functions. The antibodies themselves, again, that variable region, we also call it the FAB fragment. It's what makes that antibody recognize that antigen. FAB stands for antigen binding, okay? So the FAB part of it is the antigen binding part of it. It's what makes it really an antibody. The FC is that constant fragment that has to deal with the function of that antibody, okay? Some functions of antibodies include opsonization, serving as a molecular handle for phagocytes to grab onto. We learned that complement can do that, but so can antibody. An antibody can also activate complement pathway, right? So there's a lot of feedback loops and stuff in our immune system. Once we activate our immune system, we want all of it on board, but some of it needs to be regulated because it can be so damaging, okay? We're going. Okay, so here are the 6 functions of antibody. Opsonization is this box here in the middle. Opsonization is providing molecular handles to grab onto, okay? We can also just coat our bacteria so they can't interact with each other or with the host cells or anything like that. We can specifically bind to the spikes on viruses and block them from docking with the host cell so they can't get in. It's more specific there. We can cause our pathogens to be stuck together by agglutination. It's actually how we look for blood types too, is by agglutination. So we can cause blood cells to agglutinate with the antibodies too. We can cause complement to get activated. Remember, that's the classical complement pathway. We activated complement and it creates the membrane attack complexes to poke holes in our pathogen. We can also have antibody that binds with toxins produced by the bacteria, like we have with tetanus toxin. So we block the toxin can't have action anymore. We're protected from the action of the toxin. So those are the 6 functions of antibody. They do quite a lot, right? There are 5 types of antibody. The first one, so here's the deal. The one found on the B cell as the receptor is IgD. The first one that you secrete is IgM. That's always the first one you make. All right, it is this pentamer, it's the snowflake looking one. We secrete that first. It has limited ability. It's not as functional. When B cells are first activated, they make IgM immediately. Then they go through class switching. So that's switching from IgM to either IgG, IgA, or IgE. What determines what we're switching to? Cytokines do. So depending on what signals we're getting from an infection. So now when I tell you it's the tip of the iceberg, we're starting to see really it's just the tip of the iceberg. What cytokines are made, when, and how, and what do they signal in the cell, and what genes are being activated. That's real immunology, okay? But B cell will get signals, and based on what signals it's getting, it'll switch from IgM to these other guys, okay? IgG is your main workhorse. Generally speaking, in almost most types of infection, IgG is the #1 that's produced. So we'll go from IgM, class switch to IgG. 80% of your antibody produced will be IgG at that point. IgG is easily the most important one in your bloodstream for protecting against infection in the tissues and stuff like that. IgA is made in the mucosa. It's focused in the mucosa, okay? IgE is associated with parasitic infections, and because we don't have many of those in first world countries, this is usually associated with allergies. And your body wasn't trained not to make IgE, so it just does to like pet dander and things. So that's where IgE comes into play. We'll talk about IgE and how it's related to allergies in chapter 17. Okay, so those are the 5 types of immunoglobulins. Here's a question, we're not going to do it. Cool. All right. Titer measures how much your blood has of certain antibodies. And we can look for certain types of antibodies. We can look for IgM. We can look for IgG. And if you have a lot of IgG, you've probably been dealing with an infection for a little while. Otherwise, if we see a lot of IgM, it's probably you just saw that infection. You just started making IgM. That can tell you things about the progression of the infection. Your memory cells. Once you switch from IgM to IgG, your memory cells remember IgG. And that way, the next time you see something, you make IgG right away. You don't have to wait on it. And it's super effective. You make a ton of it right away. You don't have to wait on proliferation and differentiation and class switching. Your memory cells remember, and they can start cranking it out, okay? Titer can also be related to how recently you saw a thing. But now we're also seeing, if we have to see the pathogen outside, have a macrophage eat it up, present it on MHC2, find the T helper cell that sees Barry's eyeball, right? Make sure we're getting the right activation for that T helper cell, proliferate it, differentiate it. And then that T helper cell has to find the B cell that also sees Barry's eyeball and activate it and proliferate it and differentiate it. And then we get antibody. There's obviously a delay between seeing that antibody get the antigen get into the body versus when we make the antibody. And it's like a week or so. And it depends on how soon we get to that antigen based on your lymph and your filtering and all of this stuff. So that's the latent period. So the whole reason that I got sick with the chickenpox is because I didn't have antibody immediately available to fight against it. I had to wait and have the illness and rely on my innate system to try to fight off what it could until my body made antibody to clear it out. Now that I have antibody, the next time I see somebody, next time I see the chickenpox virus, varicella zoster, my body will just bam, IgG right away, so fast that I never get sick. That's the secondary immune response. So the primary response is latent because we're relying on all of that to happen. And it's a lot. But the secondary response is immediate. We don't need macrophage to activate a T helper to do blah, 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 blah. We can just make the IgG. Make sense? That's the goal of vaccination as well. It activates the exact same response, but we have already primed your body to react immediately. Instead of you having to get the disease, you can just make the IgG. That's the goal of vaccination. Vaccination is not perfect, and there's different types of vaccines that we can have as well. If you have immunity that you develop and you make your own antibodies to something, that's active immunity. If I gave you the antibodies that somebody else made, that's passive immunity. So you give your baby your antibodies when you breastfeed them. That's passive immunity. That's a natural passive immunity. You go to a hospital and I give you immunoglobulin because you can't make your own antibodies or because you have rabies. Not have rabies, okay? You got bitten by a dog that we think has rabies, and we want to give you antibody against rabies. That's immunoglobulin infusion. That's a passive but artificial immunity. So you're not getting it naturally. We're artificially getting it to you through medicine. When I got the chicken pox, I developed antibodies to it naturally from a natural infection. When you got vaccinated for varicella zoster, you got an artificial but active immunity. So that's the differences between these. We're tracking with that? All right. Okay, so it still activates the same pathway when you get vaccinated. We can use whole organisms or we can use parts. We can use live or we can use dead organisms, okay? We can, if we use parts of them, we can use the spike proteins, we can use other surface molecules, we can use mRNA that codes for the surface molecules that your body will then make it, and all sorts of stuff to make our vaccines. Boosters are necessary because your memory cells are not eternal. They will eventually, like, I'm not getting signaled anymore, and your body will let them die out. So boosters are there to make sure, hey, no, don't go to sleep. We want to keep you active. We want to keep you around for just in cases. And that's the idea with a booster vaccine. Make sure that we're stimulating the body enough to keep those guys alive and keep them awake. How do we vaccinate people? Subcutaneous is under the skin, but not quite into the muscle. So into the dermis typically. Intramuscular, we're actually injecting into the muscle. Whereas intradermal, we're usually excoriating. So we're causing scratches or something like that. And that's part of how you deliver it into the skin, scratching it onto the surface of the skin or into the muscle determines what types of immune responses you might get. And so for some antigens, you might want different types of reaction of different cells and all sorts of stuff. So what types of vaccines do we see with intradermal? That is the smallpox vaccine. It's really the only one that we have that's intradermal like that, okay? Oral and nasal vaccines, we don't see them a lot in first world countries, but they are easily delivered in third world countries. That's vaccination efforts for things like polio and stuff like that, because they don't rely on needles. So we don't have to switch needles between person to person. We can just deliver it orally or nasally. There's a little bit of an issue with stability and stuff like that, and they're a little less effective, but it's still better than not vaccinating the populations.
Is there a flu vaccine that they do nasally?
Yes, if you are allergic to eggs, then you will get the live attenuated flu vaccine.
Doing that.
And so sometimes they'll choose that. There's like going to be a risk for a large population of being allergic to eggs or something like that, which is the case if they're doing like mass vaccination efforts like that. And the kids can't tell you that they're allergic to eggs. They're usually not smart enough. So we would, right though, but like they don't know that they should tell people stuff like that. So we would usually just, for that type of a group, just give them the oral one. But if you're an adult, we can ask you, are you allergic to eggs? No? Okay, we're gonna give you the shot.
But if they didn't know, then is that Guillain-Barré?
I mean, so typically with that, we're talking about anaphylaxis as a result. So you'll have an anaphylactic shock reaction to the egg that's present in the flu vaccine. So that's not the flu virus or the vaccine that's causing that. That's a reaction to the egg. And it's an anaphylactic allergic response to that. So we'll learn about anaphylaxis in chapter 17 more. But those are risks with stuff like that. Guillain-Barré can happen as a result of the immune response being triggered and then the tissues near their immune response being aggravated from inflammation. And Guillain-Barré is a nervous, peripheral nerve disease, basically associated with that inflammatory reaction. It's extremely rare in vaccines, but it does happen, right? What is the likelihood that you're going to have severe damage from having the flu, though? Much higher than having the likelihood of Guillain-Barré from having the vaccine, just to be clear. But yes. All right, adjuvants are things that we add to the vaccine to be sure we're getting the right response. So if you want a CD8 memory response, then you might add adjuvants that skew towards CD8. If you want antibody response more, then you might add adjuvants that increase that. To put this into perspective, again, if you have the measles, actual sick with the measles, okay, the likelihood that you're going to develop memory to the measles is actually slim. Unlikely that you'll get memory from the measles because the measles infects your memory cells. and kill them. But if I give you parts of the measles virus and give you adjuvants to help trigger memory development, now you have one of the best and most effective memory-based vaccines against measles. So now you can just make IgG in response to seeing the measles without having to rely on anything else. And that virus will never get a chance to infect any of your cells. So that's the difference between these. Now, are all vaccines that effective and that reliable? Absolutely not. We wish and we try, and constantly science is trying to develop more and more effective vaccines that are less and less likely to have side effects with them. But it's just not a perfect system.
So it's
More-
like the shingles shot?
Huh?
Is it like the shingles shot too, 65? And you
Yes.
don't ever have to take it ever again?
Yes, pretty much.
And
So
then-
MMR, typically for most people will maintain, if you've gotten your initial shots with MMR and you had your boosters when you were a kid, most people maintain an effective lifelong immunity. Now, if you're going into healthcare, now some of you guys are probably going to healthcare and you're having to get your titers done
Mhm.
for the nursing school program, they want you to prove that your memory is strong enough. And if it's not, they'll give you another booster. Okay? So that's really what it is. They just want to make sure that your memory is strong enough. Again, not for you, so that you don't get patients. That is what they're worried about. Common side effects with vaccines almost always rely on the fact that you're having an immune response. So you'll have local pain and soreness and stuff like that. Again, we're initiating an inflammatory response in your body intentionally. So you'll have pain and things like that from the vaccine, or you'll have short-term flu-like symptoms. You get any vaccine, you can end up with flu-like symptoms from it. Are you sick with the flu? Are you contagious? No, 0% chance actually. No. Impossible, actually. Like if you get the flu shot, that is dead, killed virus. It's impossible for it to replicate. It's not alive. It's as dead as you would be. You're not coming back to life out of the grave, right? It's as likely that you'll come back to life out of the grave as that virus is likely to cause the flu. But do you have the signs and symptoms of the flu? Yes, because we're literally intentionally triggering your immune reaction to it. And 99%, this is the thing I think that people get lost on. They're like, then why do I feel like I have the flu? 99% of all of the illnesses that you have is your body's reaction to the pathogen. It's your inflammatory response. It's that cal or dole or tumor. It's that being triggered from your innate response. Now, if the organism is found in your skin more, then you'll see rashes and things like that from the inflammation. If it's found in your inner tissues, you'll feel malaise, fatigue, soreness, and things like that. But that's your immune system making you feel like crap, not the virus. Now, if you have Ebola, what's making you bleed out? The virus. Okay, I'm just saying. So it's not 100%, but most of our immune responses to normal infections is caused by your immune system, not by the virus causing the issues. Are we good? Are we tracking that? Okay, risk of anaphylaxis is the biggest, most serious side effect associated with these things, and it's usually some ingredient in the vaccine that the person is allergic to that can cause anaphylaxis. Again, you can't get the infection of the flu from the flu vaccine. This is just caused by triggering your immune response. Number two, mercury in the measles vaccine does not cause autism. There was a paper published on that was retracted, and the person lost their medical license and is now hired by RFK to be one of our health advisors for autism. But it was false. It was falsified. It was paid to be falsified for the favor of the pharmaceutical companies at the time, okay? But it has been proven multiple times that the mercury that might have been used to create the vaccine is not what is linked to autism. Now, is there mercury used to make some kinds of vaccines? Yes, there is. Is it still present in the vaccine when you get it? Not usually. And if it is, it's at small amounts. You're like, well, that's still enough. Like, I'm still worried about that. Why are you drinking Coke or eating food that you bought from the store then? Because that all has more mercury in it than you will ever see in all of the vaccines that you'll ever get in your entire life combined times like 1,000. Like one Coke will have more mercury in it. Be real about what your issues are and what we're facing in life. Be educated. If you've ever ate a piece of fish, it probably had 10,000 times more mercury than you'll ever see in any vaccine ever. Be real here. You want to cut out mercury? Cut out mercury. Don't let me stop you. Mercury is bad for you. Is your issues with mercury and health effects caused from your vaccines that you got? Absolutely not. And your body is built to deal with these things too. Okay, we clear? Any questions? All right, that's it for this chapter, I believe. Please help. Yes, okay. All right. All right, so we will review all of the immune system stuff in the review on Thursday. Otherwise, we're gonna head up to the lab. Let's head straight up there. If you need a bathroom break or you need to go grab your lab coat, go do that. Give you guys a couple minutes to catch up. The lab should be shorter today. It was for everybody else, so I'm not too worried about it. Great job today, guys.